Okay, I think we can get started. Thanks so much for joining us today, almost a year later than the conference was originally planned. And what a year. Now on the second day of a new beginning, let's all hope this year will be much better in every aspect than last year. I'm Xiaoxia Cui. I'm the director of the Genome Engineering and iPSC Center here at WashU. Before we go straight to the full agenda we have today, I want to thank Candace Farmer for all her help organizing this meeting, and Jonathan Goldstein at WashU IT for technical support. So any issues you have with the meeting, please use the chat box. Candace and Jonathan can take care of you. And please ask questions after each talk in the Q&A box and mark who the questions are to. I'll ask the questions for you. Unanswered questions with answers will be posted on our website after the meeting. The other speakers, I just want to remind you that you have access to the microphone on both days. Just remember to unmute yourself before speaking. The talks are being recorded and will be posted on our website with the speaker's permission. Without further ado, let's start with our keynote speaker, Charlie Gerspac. Charlie really doesn't need an introduction, and I actually think a lot of you came here because of him. At least that's how we planned when we advertised the conference. I think it worked. What I just want to point out one thing that I saw on his website, that under research interests, he lists gene therapy, genomics and epigenomics, biomolecular and cellular engineering, regenerative medicine, and synthetic biology. That sounds like everything in biomedical research to me. That's how I feel about Charlie's research all along. So his title today is Decoding and Programming the Genome with CRISPR Technologies. Charlie, can't wait to hear this one.
Thanks so much. Go ahead and share the slides. Thanks for the really kind introduction, Xiaoxia, and thanks so much for the opportunity to come and present. Obviously, I'd much rather prefer to be there in person with all of you, as much as I appreciate giving this talk in my slippers. But hopefully at some point in the near future, we'll all be able to get together. So yeah, I mean, I think that obviously it's a pleasure to give this talk and to this audience. These are disclosures that I have to disclose before I go ahead and get started on the research side. But as I was thinking about what would make sense to talk about today, it was a little challenging because there is so much exciting stuff going on in the field, and a lot of different timely topics, and the field is moving so quickly. And even this is a timely meeting with the Nobel Prize for CRISPR in just a couple months ago. And so in thinking about what maybe to focus the talk on, I was remembering this quote from Dr. Jennifer Doudna after being awarded the Nobel Prize about what she thinks the next big, where the next big CRISPR advances will be. And just to paraphrase, it was, her answer was using CRISPR not to edit genomes or make permanent changes, but instead to regulate them and control levels of proteins produced from different genes in order to control cells. And I was really excited when I saw this quote following the presentation of the Nobel Prize, given that it's an area that we've had a lot of interest in the last several years. And so even though we do a lot of work in the area of gene editing for functional genomics and treating genetic disease, I decided that today I'd focus the talk in this area of epigenome editing and targeted gene regulation. And even stepping back from CRISPR a second, we think about the epigenome editing technologies very broadly, with its most generic form being some type of DNA binding domain shown here in the middle, which can be targeted to a specific DNA sequence and then is either attached to directly or can recruit what we call the effector domain that modifies the epigenome or gene expression. And so if you can actually go in and design one of these tools to go in and target one specific site in the genome using a DNA binding domain, such as an engineered zinc finger protein or a tau effector or your favorite CRISPR-Cas system, then it can be imagined that could be very powerful for lots of different applications across many different areas of biomedical science and potentially medicine, as Zhaozha indicated in the introductory remarks. And so as you go around the periphery of the slide, these are things where we've been really excited to apply these tools in recent years, starting in the top right, the idea of going in and changing an epigenetic state at one site in the genome and one site only, very specifically allows you to better understand the kind of cause and effect relationships of epigenetics. While the catalog of different epigenetic marks seems to be growing by the dozens all the time, different types of DNA methylation changes, different types of covalent histone modifications, we know very little about what any of those do and whether they are the cause or the effect of changes in gene expression or chromatin structure. And so this allows you to go in and test some of those hypotheses directly. We've also used these for high throughput screens of the epigenome. We'll talk more about that in a little bit. They can be used as components in gene circuits and synthetic biology. They can also, and we'll talk maybe a little bit this at the end about how these types of tools could be used directly for gene therapy by, rather than delivering a particular gene, actually delivering the machinery that can go in and regulate genes in their endogenous position. And then I'm going to spend most of the talk today is talking about cell programming. And so how can we use these tools to specify cell lineage and phenotype? And so while this cartoon presents this as a generic DNA binding domain that, as I mentioned, could be any type of DNA targeting molecule. Like many of you in recent years, our favorite types of DNA targeting tools have been the CRISPR-Cas systems, and in particular, Cas9. And as many of you probably know, while Cas9 is most famously used as a gene editor, where the Cas9 protein binds to its guide RNA and finds a target site in the genome, where it then uses its nuclease domains to cut the two strands of DNA, inducing DNA repair pathways that can be then used for gene editing. We actually strongly believe that that's kind of just the tip of the iceberg of what these technologies can do, and that what's most exciting about CRISPR technologies is not gene editing and not cutting DNA, or even necessarily changing DNA sequence, but is really the ability to take a protein RNA machine and target it to one specific site in the genome, and then recruit any activity that could change genome structure or function at will. And one of the areas that we've been most interested in is what I introduced as Epigenome Editing. And this can be easily done by introducing 2-point mutations into the Cas9 enzyme, knocking out its two nuclease domains. So it still binds this Cas9, modified Cas9 protein with the 2-point mutations, will still bind to its guide RNA. It'll still find its target site in the genome, but it no longer has the ability to cut. So now it just becomes a protein RNA scaffold for adding additional functionalities onto. And as shown here, that effector domain could be one type of effector domain could be something that modifies local epigenetics and gene expression. And so there's been many people in the field advancing these technologies since concurrent or shortly after the kind of discovery of using CRISPR for editing applications outside of its natural context. And one of the areas, and we're going to hear, I think, from many of those people over the course of this meeting, but I think that one of the areas that we've been particularly interested in from the early days is making these dead Cas9, referring to the nuclease inactive Cas9, or dCas9 for short, based transcriptional activators. And one of the reasons for that is that there's lots of ways to potentially get rid of a gene or knock down a gene or block the product of a gene, whether it be the RNA or protein. But there's really not too many ways that you can turn on a gene from its natural position in the chromosomal DNA. And so that's where we have put a large amount of our attention from early on. And one of the activators, domains that we use is something called VP64, that is really just a generic acidic activation domain derived from a viral protein that can recruit RNA polymerase and other transcriptional initiation machinery. And so the early work that we did back in 2013 was to take this dCas9 VP64 and a combination of guide RNAs and target it to a promoter of a gene that was basically undetectable in the cells that we were working with. And we showed that we could, in fact, activate this gene to robustly to to very detectable levels. In those early days, we had to use a combination of guide RNAs targeted to a single promoter. Now, with a variety of next generation activator domains that we and others have developed, you can actually accomplish the same result with just a single guide RNA. But while we were really excited about the idea that we could turn on a gene from its natural position in the genome, the real question was how specifically were we doing this, right? So if the goal was to just turn on this one gene and leave all other genes alone, that was going to be a tall order when we were taking this very potent transcriptional activator and overexpressing it in the cell line. But we were really pleasantly surprised to see when we took the same cells and ran RNA-seq that the only genes that were significantly upregulated were the 4 isoforms of this IL-1RN gene that we targeted in this experiment. And so that got us excited that we could do this very specifically. It also opened up another question that we didn't think about until after we had done this experiment, which was, if this gene is off and then presumably repressed in some type of silent heterochromatin state, how is it that CRISPR can get in and access these genes and turn them on? And we were building data sets around turning a number of different genes on and a number of different cell types, but there was also literature accumulating indicating that Cas9 or dCas9 did not, preferred open chromatin to closed chromatin. And so we then later went back to reevaluate this experiment to understand what was going on with chromatin state. And in fact, using a method called DNA-seq, which is a genome-wide method for assaying chromatin accessibility by treating chromatin with DNase, it's only cleaved in the open regions. We could, profile this promoter of this kind of model gene that we were looking at, and see that in fact, in all of our control samples, there was no DNase hypersensitive site here, meaning that this was closed chromatin, inaccessible to the DNase-1 enzyme. And that was true in the starting cells. It was also true if we delivered the dCas9 with or without the activator domain with control guide RNAs targeting another region of the genome. However, when we delivered the correct guide RNAs targeting this promoter site, we see that we actually created a new chromatin accessibility site. And there were a couple of things that were interesting about this. One was that this shows, I think pretty conclusively in our mind that the dCas9 does have the ability to go in and access regions of the genome that would otherwise be categorized as closed and inaccessible, and likely displace nucleosomes to create these types of accessible sites. The other thing that was really interesting to us was that it had nothing to do with the VP64 domain. The size of the accessible site was equivalent with either dCas9 alone or dCas9 VP64, even though we only saw changes to gene expression when we included VP64. So this indicated that dCas9 should be able to access these heterochromatic regions, regardless of which effector domain you put on it, potentially, and that you can independently tune chromatin structure, meaning opening up these heterochromatic sites while also independent from actually turning on gene expression, which I think expands the toolbox available for manipulating the epigenome and chromatin structure. So with these experiments in hand, knowing that we could turn on genes that were otherwise off, knowing that we could do so very specifically and target one gene without manipulating any other transcripts in the genome, and then knowing that we could access heterochromatin and open it up, we started to think about what the world of applications might be for these tools. And one of the areas that we got most excited about was programming self-phenotypes, which was really inspired by this concept of the Waddington landscape that I'm sure many of you have seen. And so briefly, this marble at the top of the landscape is a pluripotent or totipotent cell that could become anything. And as it falls down the energy landscape and falls into one of the different valleys, that represents kind of terminal cell differentiation or specification decisions. And while I think for decades, this was kind of represented as a irreversible process where these hills in between the valleys represented barriers that prevented one differentiated cell type from becoming another. And the metaphor, of course, being that those cells represent the epigenetic barriers that prevent these cells from converting state, given that we know the underlying genome sequence is the same in all of these different cell types. We now know from the reprogramming field that in fact this is a reversible process, that with things like overexpression of master transcription factors, you can not only reprogram differentiated cells to pluripotency, but it can also directly convert cells between fates, directly traversing these epigenetic barriers in the Waddington landscape. Now, when we started down this area, several years ago, the vast majority of the work in this field was done with overexpression of these master lineage-specific transcription factors that have been well documented to bind to tens of thousands of sites across the genome and make many different changes concurrently. And in fact, it had also been shown that in these kind of reprogramming protocols or direct conversion protocols, where these master transcription factors were being overexpressed, not only were they going to tens of thousands of sites that they would naturally go to in the cell type that you might be trying to create, but they were going to many other sites as well because they were being overexpressed at physiologic levels. And so weak binding sites that they wouldn't normally be located at could readily be detected by assays such as ChIP-seq. And so the point of that is that there's many different changes being made to the epigenome, genome-wide, some natural, some not so natural, even though some of those unnatural changes might be critically important to the actual getting the reprogramming accomplished. And so only with the emergence of these types of targeted epigenome editing tools did we realize you could start to ask the question of, okay, what are the necessary and sufficient epigenetic perturbations to convert cells between phenotypes? Rather than making tens of thousands of changes across the genome, can we make just a few to understand what the important pieces are that define cell lineage specification? And so we've done this now with many different cell types, but one of the first areas where we wanted to explore this question was the conversion of fibroblasts into neurons. And this was really inspired by work that Marius Vernick had done, showing that with this combination of three transcription factors, neuronal transcription factors, BRN2, ASCL1, MIT1L, what he called the BAM factors for short, that either in mouse or human fibroblasts, overexpression of these could convert fibroblasts to neurons. But he also showed that these, as I mentioned, were binding to tens of thousands of sites and making many changes. So Josh Black, a grad student in the lab, asked the question, well, what if rather than overexpressing these master transcription factors from viral vectors, we just delivered a DCAS9 activator that went into the promoter that encodes these genes naturally, these three master transcription factors as a starting point, and turned them on. And so naturally in fibroblasts, these neuronal transcription factor genes will be off. The question was, could we deliver the activator and turn them on? And then critically, could we do so in a transient way, where the DCAS9 activator is present only transiently, changes the epigenetic state at those target promoters in the endogenous chromosomes, and then goes away, degraded or lost, and then ask the question of, is it a stable new phenotype that can maintain itself? And the way that we did this was actually by transient transfection of plasmids into the mouse embryonic fibroblasts that we were working with, and knowing that the plasmids would degrade if we lost after a couple of days. And so the experiment that we did was to either do the traditional method of encoding the BAM cDNAs on these plasmids and transfecting those into cells, or encoding on the same plasmids the DCAS9 transcriptional activators, or what's been called CRISPR-A, along with guide RNAs targeting the promoters of BRN2, ASCL1, and MIT1L and their endogenous chromosomal position. And so what we saw was that, of course, so the control plasmids, there's no detectable levels of these neuronal genes because these are fibroblasts. Now, if we overexpress the cDNAs from plasmid, we see very high levels, and note the log scale here, which makes sense since we're just blasting expression of these things as high as we can from these transfected plasmids. The good news was that we can also turn these genes on from their chromosomal copy using the CRISPR-based activator, but as you see, it's an order of magnitude or more less expression than what we get when we just blast them from the plasmids. However, this is looking at total expression of the cDNAs. If we change the primers so that we're only amplifying the transcripts that are getting made from the chromosomes, from the natural copy of the gene, you see that everything in the red on the left translates over to the right, because the only place where the transcription of these genes can come in the CRISPR-A samples is from the endogenous copy. But what we also notice is that the blue bars get much, much smaller, which indicates that even though you get very high levels of these factors being made when you transcribe them from transfected plasmids, you're actually, those same factors are doing a very poor job of turning on their corresponding endogenous copy in the chromosome. And so what we expected was that if this is true, then after a couple of days, once all the factors, all the plasmid gets washed out and all the overexpressed factors go away, that you might get a more stable phenotype by turning on the endogenous copies, which can then continue to be expressed in the absence of the plasmid. And in fact, that's what we saw when we looked at 2-3 weeks after transfection. We got about twice as many of these stably converted cells in this neuronal phenotype, both by markers, morphology, and electrophysiological properties, when we used the CRISPR-based activation of those genes as opposed to just plasmid overexpression. And then finally, to kind of further test our hypothesis that this is because we're changing the epigenetic state of these genes at their endogenous promoters, we did a chromatin immunoprecipitation for 2 different active histone marks, H3K27 acetylation or H3K4 trimethylation, at the promoters for all 3 of these genes. I'm just showing ASCL1 here. And what you see is enrichment of these activating histone marks only with the CRISPR-BAM treatment. And so, this further supports the idea that by flipping the epigenetic state at the endogenous copy, we're getting more efficient and more stable conversion of the cell phenotype. So that was work that we'd done a couple of years ago, just testing the idea of can we use these to program cell phenotypes. And we wanted to build on this idea to either convert cells like fibroblasts to many other types of cells, or even to direct the differentiation of pluripotent cells into specific lineages or phenotypes or subtypes. And we realized that that project, we had the benefit of what Marius Wernig had already shown us. He had already pointed out these are the 3 master transcription factors that you should turn on if you want to convert fibroblasts into neurons. But we also realized that there were many other factors that, or there were many other cell types that we'd be interested in creating where we didn't know what the master transcription factors might be a priori. And so we then sought out to take advantage of CRISPR-based pooled screening, building off of work that people like John Dench and many others had done in years previous. We want to take advantage of this idea to identify the factors that would allow us to convert one cell type into another. And so the way that we did this was to start out with a human iPS line, because we wanted to see, can we make different cell types out of these iPS cells, that we had engineered first to express the DCAS9 activator, and then, but also where we had knocked in an mCherry downstream of the TUBB3 gene. And TUBB3 is a marker of neuronal lineage specification. So any cell that starts to move down the neuronal lineage is going to turn on TUBB3. And so in these clonally derived engineered cells, those cells would then also fluoresce red. And we can then use red fluorescence as an indicator of neuronal lineage specification. And so we then took this engineered cell line that has the activator but no guide RNAs and delivered a lentiviral guide RNA library. And maybe I'll jump ahead 1 second to tell you about that library. What we did is we started with looking at the roughly 1,500 different human transcription factor genes. So these are human genes annotated as transcription factors. And then we pulled from a library that Jonathan Weissman and Luke Gilbert and colleagues had published previously to pull 5 guide RNAs for every single one of those 1,500 promoters that drives the expression of a human transcription factor gene. And we added some extra controls as well. So the whole library was about 8,500 different guide RNAs. And we packaged those into lentiviral in the lentiviral vectors so that every particle carries a different guide RNA. And we introduce this into cells at a low MOI in this case, so that every cell gets a different guide RNA. And so then in every one of these cells, that guide RNA will pair up with the DCAS9 activator and turn on a different human transcription factor gene. We wait 5 days, and then we sort off the red cells that are markers of having gone down the neuronal lineage specification, and then sequence that pool of guide RNAs and compare it against, either comparing the high red, the high M cherry expressors to the low M cherry expressors, or to the bulk population. And so that's what this looks like. In this case, we sorted off the 5% highest M cherry expressors and the 5% lowest M cherry expressors. And in this graph, I'm showing the M cherry, the distribution of the M cherry high guide RNAs versus the low. And what you see is we've got a number of different, everything in red is statistically significant. And so we've got a number of different guide RNAs that were enriched in the M cherry high population, indicating that those guide RNAs turned on a transcription factor gene that helped push those cells to the neuronal lineage. And so we'll focus on those first. I'm going to come back to these ones that are depleted in the M cherry high samples in a little bit. Focusing on the guide RNAs that were enriched, we can then look at those guide RNA sequences, map them back to their target gene, and then go in and validate them either by delivering that guide RNA alone or another guide RNA targeting that gene, or even a cDNA encoding the gene that we regulated. And so the good news was that when we go back and we try and validate the guide RNAs for their ability to turn on that TUBB3 M cherry reporter, we see almost perfect validation of the screen. So we feel like the screen in and of itself was very robust. Now, of course, this is telling us that those guide RNAs targeting any one of these genes could turn on the reporter. That's all that it really tells us. So we then wanted to look a little bit more carefully to see, is it turning on other markers of neuronal lineage specification? And that's kind of interesting because it's not always the case. The validation doesn't work quite as well there. You see many of the genes that turned on the reporter also turned on these other neuronal markers, NCAM and MAP2, but not all of them. So for example, SOX4 very robustly turns on the reporter, but doesn't actually turn on any other neuronal markers. And so it shows that these validations are important, and also shows that there might be a number of other ways that the reporter can get turned on other than what we were looking for. Nevertheless, we can parse through all of this. We can also look at morphologies and other markers. And the good news was that we found a number of different targets that seemed to push these cells into the neuronal lineage. Some of these were well known, like your neurogenins, et cetera. And so that was good validation that the screen worked. And some of these were less well known or previously associated with neuronal lineage commitment. And so it gave us a lot of interesting things to follow up on. But one of our primary questions was, how are these targets different from each other? So we got dozens of different targets that could seemingly push these cells down the neuronal lineage, but TUBB3 is a very early marker of neuronal lineage commitment. And so we want to know how each of these targets, what were the differences between the cells that we were generating. And so one of the ways we did that was by going and individually upregulating the different targets, and then doing RNA-seq and comparing the samples against each other. So this is an example of two of the strongest hits, neurogenin 3 and ATO1. And so if we go in and activate those individually and compare their transcription profiles, and we basically take all the genes that were differentially expressed compared to the starting cell population, we see a lot of those are shared, right? So 3,000 of those genes are shared. And if we say, okay, well, what are these 3,000 genes? They're things involved with neuronal differentiation. So that's very reassuring. But that's also probably not the interesting stuff, right? The interesting stuff is, what are the genes that were only turned on by neurogenin 3 and the genes that were only turned on by ATO1? And so if we look at that more carefully, it got a little bit more interesting in that the genes that were only turned on by ATO1 and not NGN3 were enriched for markers of dopaminergic neurons, whereas the flip side of that, the genes that were turned on by NGN3 and not ATO1 were enriched for glutamatergic markers. And so this got us excited about this idea that maybe some of these hits can get us to specific subtypes of neurons and a more specific type of lineage specification than had been developed by previous methods. But then, that got us thinking even more about the fact that, in that experiment, we were only turning on one transcription factor at a time. But if we really want to get to, fancier cell types and more specific lineage commitment, we know that those types of phenotypes aren't typically governed by a single transcription factor, but typically by combinations of transcription factors. And so we wanted to go back and revisit the screen and do it in the context of combinations of factors. And so in this case, what we did was we used lentiviral vectors where each vector encodes for 2 guide RNAs. And what we did is in the first position, we put one of our strongest hits and kept that fixed. And then we cloned the library targeting all other human transcription factors into the second position. And we did this twice. We did it once where the first position was a guide RNA targeting ASCL1, and we did it again where the first position was targeting NGN3, both of those being well-established master specifiers of neuronal lineage commitment. Everything else was kind of done the same way, but the hits were very different, which was very interesting. And so now you see that everything over, so we're looking just at the neurogenin3 plus the CRISPR transcription factor library. And so everything over here was in the mCherry high population. So these would be factors that synergized with NGN3 to push these cells to neuronal commitment. And then everything over here would have been enriched in the low mCherry or depleted in the high mCherry population, meaning that those were things that antagonized what NGN3 was doing to push these cells into neuronal lineage commitment. And so if we do this type of analysis, both for NGN3 and for ASCL1, it's kind of interesting because you see that there are some factors, this is only looking at the positive factors, the ones that synergized, and you see some of these synergistic factors were shared between ASCL1 and NGN3, but some of them were specific to either one of those 2 transcription factors, falling either on the y-axis or x-axis. And so then once again, we wanted to understand, okay, how are these pairs of guide RNAs or pairs of gene targets different from each other? And we did this again by delivering them either NGN3 targeting guide alone or NGN3 targeting guide along with a guide targeting one of the other hits from the screen, doing RNA-seq and comparing the transcriptional profiles. And again, it's interesting because a lot of the genes are shared. You see here 2,500, but a lot of genes were relatively specific for particular combinations of targets. And one that stood out to us was this combination of NGN3 with LHX8. The reason being that when we did a gene ontology analysis for these 180 genes that were specific to this sample, we had got a lot of hits in the area of synaptic maturity and axon development. And that was interesting to us because we knew that one of the limitations in this field of making iPS-derived neurons is getting mature cultures. Often that, the cultures that you get from these iPS-derived neuronal differentiation protocols are very immature, or you have to wait long periods of time for them to mature. And we thought that this might indicate that LHX8 was working with NGN3 to accelerate the maturation of these cells. And in fact, one of the ways that we looked at that was a collaboration with Scott Soderling and cell biology at Duke, where he used patch clamp analysis to look at the electrophysiological properties of these cells. And in fact, that's what we saw, was that at very early time points, when we added LHX8 to the sample, we got more action potentials more often, more frequently, and in more cells. And with properties that were more indicative of mature cultures. And so, so that further got us excited about this idea of using these to get subtype, using these approaches to specify subtype, to enhance maturity, et cetera. Now, that whole approach, though, was focused on the things that were synergizing together. We wanted to go back and actually also revisit the things that were antagonizing the differentiation to neuronal commitment. And another way of looking at this is just the things that were depleted in the mCherry high versus mCherry, or either enriched in mCherry low or depleted in mCherry high. So basically these candidate negative regulators. And so looking at these here, again, you see that some of those negative regulators were shared between the ASCL1 and the NGN3 combinatorial screens, and some of those were unique. But then we asked the question, well, if if some of these negative regulators, what if some of these were already being expressed in the starting cell population, then they might actually, before any perturbation, then those would presumably be acting as barriers to the differentiation that we wanted to achieve. And so we looked at this more carefully, and in fact, that was true. Some of the strongest negative regulators of lineage specification were HES3 and this zinc finger protein that were both expressed at reasonable levels in pluripotent cells. And so that led us to ask the question, if these are acting as barriers in the starting cell population, and they're even stronger barriers when we turn them on, then maybe we can use repressive CRISPR epigenome editing tools to turn those off and enhance neuronal differentiation. And so now what we did is we used orthogonal CRISPR activators and repressors based on Staph aureus Cas9 and your conventional SP-Cas9. And we showed that with the SA-Cas9, we could actually turn off these negative regulators of neuronal differentiation from pluripotent cells. And then if we combined the repression of these negative regulators with the activation of the positive regulators, that actually we could get enhanced lineage specification. So I think that this is kind of an early proof of concept around the idea of using these orthogonal epigenome editing tools to make different types of epigenetic perturbations around the genome to kind of get a more sophisticated programming of the exact cells that you want. Okay, so I think that a lot of that, moving to the kind of another short story or two about this area that we've been working on. The whole idea of programming neuronal differentiation, we were interested in at first for proof of concept that, if we wanted to show that we could use these epigenome editing tools to move cells around the Waddington landscape, then either conversion of fibroblasts to neurons or differentiation of pluripotent cells to neurons was a good test bed for demonstrating that. We've also been really interested in this idea of making these very mature neurons or these subtype specific neurons for things like drug screening or disease modeling, in a dish for types of human neurodegenerative or neuropsychiatric conditions that are otherwise difficult to model with existing protocols. But in addition to all of that, we were also really fascinated by the idea of how we could use these tools for regenerative medicine and cell therapy. And could we actually use this to generate cell types that could be used in a therapeutic context? And one of the areas that we've been working in is skeletal muscle, treating skeletal muscle disease and skeletal muscle regeneration. And one of the reasons for this is just we find skeletal muscle is a really interesting tissue for testing lots of different things. It makes up most of a large amount of our body mass. There are genetic diseases, there are diseases of aging, other weakness. It's a regenerative tissue. It's a tissue that has endogenous stem cell population. And so all of these things make it a really interesting test bed for evaluating gene editing tools to treat genetic disease, or gene regulation tools for manipulating diseases of aging and regeneration or degeneration, and also for generating stem cells that could be involved in the regeneration process. And so in particular, the stem cells of skeletal muscle are called satellite cells. And so shown in this cartoon in the top right, muscle, mature muscle tissue is made up of these highly multinucleated muscle fibers. And these satellite cells are the stem cells that are mononuclear, sit on the outside of the muscle fiber. And in response to tissue damage, as shown in this pathway down here, these satellite cells will asymmetrically divide and then begin to differentiate and ultimately fuse into the muscle fiber, adding new nuclei to the muscle fiber and participating in membrane repair. So we were interested in the idea of taking advantage of the satellite cell population for muscle regeneration. And we also knew that the master transcription factor that's been known to govern the satellite cell fate is called PAX7. And there had been previous protocols that had used PAX7 overexpression to drive multipotent or pluripotent cells into a satellite cell-like phenotype. But given the work that I've shown over the last 40 minutes or so, that we think that there's a lot of advantages to turning on the endogenous copy of the gene, we wanted to explore this idea in the area of making satellite cells from human iPS cells. So the way that we did this, we took a couple of lentiviral vectors, and one of them encodes the guide RNA, as well as the tetracycline inducible transcriptional activator domain, because we wanted to be able to drive the expression of the DCAS9 activators or cDNAs in a TET-inducible way, so that if we deliver tetracycline or doxycycline, we can turn on these factors from the lentiviral vector, but then we can also take the doxycycline away and have the expression decrease, and we can see, you know, how well that new phenotype or change in gene expression is maintained. And so this TTA governs the expression from two different, either one of these two vectors. One of them is a TET-inducible vector that encodes the DCAS9 activator, as well as an mCherry marker. And then the other control sample is just encoding the PAX7 cDNA, so that we can compare again cDNA overexpression to endogenous activation. And we used a protocol with these iPS cells that was adopted from several other studies by other groups that had shown this combination of small molecules could help in this given kind of dosing regimen, could help to push iPS cells into a mesodermal lineage. And so we started with the iPS cells, transduced them with these lentiviral vectors where they all got the R33a vector, but then they got either the DCAS9 activator or the PAX7 cDNA. In this case, the guide RNA obviously targets the PAX7 promoter. And then we ran the cells through this protocol, and then we could sort the cells to make sure that we got a pure population of lentivirally transduced cells, and we used the mCherry marker for that, and that's shown here. And we were confident that this sorting to get a relatively pure population worked well, because when we then stained for PAX7 in the cDNA samples, we got 90% PAX7 positive cells. And in the case with the DCAS9 activator with the guide RNA, we actually got 98% PAX7 positive cells. So we had, at the start of the experiment, we had nice pure populations expressing PAX7 in both conditions. Interestingly, the cells that were where PAX7 was turned on from the endogenous locus with CRISPR-A were actually more easier to culture and were more expandable than the ones overexpressing PAX7. This suggested us potentially that the overexpression of PAX7 to supraphysiological levels was maybe pushing differentiation of these cells too far, or otherwise giving some type of aberrant signal to these cells. We also wanted to make sure, of course, that PAX7 was getting turned on and maintained. And this looks similar to the results that I showed earlier with the neuronal differentiation, that if we look at total PAX7 levels, we see that they're very high, either when delivering the cDNA or when turning it on from the endogenous locus. But if we look at endogenous levels, it really only gets robustly activated with the DCAS9 activator. We also learned along the way that there's multiple isoforms being made from the PAX7 gene, and we'll actually come back to that in a couple minutes. So these cells that we were turning on PAX7 were also turning on other markers of the myogenic lineage. 3 of those shown up here at the top, down here at the bottom. We saw much more kind of uniform and robust shifting of the whole population to turn on these satellite cell markers when we used the CRISPR-A based approach as opposed to cDNA. And then finally, the real reason why we did this experiment was to explore whether these types of programmed muscle progenitor cells could be used for muscle regeneration. And so for this experiment, we actually damaged the muscles of immunodeficient mice so that they're immunodeficient, so we can transplant these human cells. And then we took these engineered cell types and injected them into these damaged muscles, and then waited 4 weeks and then harvested the muscles to look for integration into the muscle and participation in muscle repair. And one of the ways that we can look at this is by staining specifically for human dystrophin on the outside of these muscle fibers. And what you basically see is that while we can pick up a couple of human cells here and there when we overexpress the PAX7 cDNA, or when we kind of deliver control cells, we only robustly see human dystrophin expression, meaning cells that had grafted into the muscle fibers and participated in repair in the samples where we use the DCAS9 activator to turn on PAX7. And so again, I think this speaks to additional evidence that these types of tools can be advantageous for programming specific cell phenotypes, both in disease modeling context, but also in regenerative medicine context. Importantly, once we transplant those cells, they no longer have the doxycycline. And so they're only staying in that new phenotype if it's endogenously maintained and autonomously maintained in the absence of the CRISPR activator. And we kind of further evaluated that here, where we see in the presence of dox, you have widespread PAX7 positive cells. And then if we take the dox away, we see differentiation of these cells and upregulation of myosin heavy, muscle specific myosin heavy chain. And we see that, again, that's much more robust when we've turned on PAX7 from its endogenous locus than when we overexpress the cDNA. This further supports this idea that we can use this dox-inducible system to show stable conversion of the epigenetic state of these cells. You see, when we give dox, either when we treat with the DCAS9 activator, we see the activator being made. When we take the dox away, the activator goes away. However, if we look at PAX7, what you see is that PAX7 is present from the cDNA when we give dox. It goes away when we take the dox away, but we do see some amount of PAX7 still maintained when the CRISPR activator goes away. Again, suggesting that once the CRISPR activator flips the epigenetic state of these cells, that the activator can go away and that epigenetic state can be maintained. And that was, again, further supported by looking at the chip using chromatin immunoprecipitation and qPCR to look at these activating histone marks at the PAX7 promoter. And whether it's H3K4 trimethyl again or H3K27 acetyl, we see that even after 15 days after we've taken the dox away and the Cas9 activator is gone, we still see enrichment for those marks. And then finally, we continue to think about why, what is the mechanism or rationale for why endogenous activation of the gene can be superior to overexpression of the cDNA. And one reason is obviously the dose, that you're giving very high levels of this transcription factor when you overexpress it, and thus it's probably going to other sites where it doesn't naturally belong and creating aberrant effects across the epigenome. But another factor is the idea of all of the biological information that's encoded at the natural locus. And some of that biological information are things like enhancers and other distal regulatory elements, other parts of the transcript like introns and 5' and 3' UTRs that contain things like microRNA target sites that can be regulated by other signals in the cell. And then also the multiple isoforms that can get made from an endogenous gene via alternative splicing that you would not see via overexpression of a single cDNA. And as I mentioned, we learned along the way that there's these two different A and B isoforms that get made from the PAX7 gene. There's actually a different C terminus on each of these proteins that get made from these two different transcripts, as well as different UTRs that contain different microRNA target sites. And to look at this a little bit more carefully, we overexpressed either the A or B isoform, and again, compared to that to CRISPR activation. And it's interesting to us that they all look very different from each other, and that we basically got the best enrichment of myogenic and satellite cell markers when we turned on the endogenous locus. And so we think that in addition to dose, one contributor of this is going to be the different proteins that get made. So to just talk quickly about where this is moving forward, we're really interested in not only enhancing the types of cells that we can program ex vivo, but also moving some of these tools for cell programming in vivo. And just to give some teasers for that, one is we have shown, and many other groups have also shown, that these same types of epigenome editing tools can be encoded on viral vectors and delivered in vivo for gene regulation. This was not necessarily a cell programming experiment, but this was a study we published a couple years ago showing that we could encode CRISPR repressors on AAV vectors, target them to the PCSK9 gene, deliver them to the liver, and get lowering of PCSK9 in the serum that then had a corresponding decrease in LDL cholesterol levels, which is a potential therapeutic approach that's being explored for hypercholesteremia and cardiovascular disease. But knowing that we can do this in vivo, we're interested in how do we advance this to doing both complex cell reprogramming in vivo, potentially doing this with orthogonal epi editors, but also doing in vivo pooled screens where we can do the same types of screens that I presented previously. Now, delivery of these epi editors is always the obstacle to these types of experiments. And so for that reason, we've developed some of our favorite CRISPR-A and CRISPR-I transgenic mice. These are now available at JAX, or we can share them directly through Duke. So if these are of interest to you, similar to the Cre-dependent Cas9 mouse, we made them based on the same model, but either encoding a dCas9 crab for CRISPR repressor or a dCas9 P300 is one of our favorite epigenetic activators. And the structure of the locus follows very similarly to the Cas9 mouse. And this is just showing that we've used these CRISPR-A or CRISPR-I mice in a variety of different contexts where we've either crossed the Cre genetically or delivered the Cre virally. And we've done this both in vivo and in cells that we've taken out of the mouse. We've shown activation and or repression in the liver, brain, heart, muscle, and in immune cells and fibroblasts that we've, and I think myoblasts that we've isolated from the mice. And so we're hoping that this will be a really useful set of tools for the community. And if that's of interest, we hope that you'll acquire those. So just to summarize, the hope here was to talk about where some of these types of CRISPR and genome engineering technologies might be going in terms of above and beyond gene editing and changing underlying DNA sequences. There's lots of applications for these as I introduced, but one of our favorite is programming self-phenotypes and using them for screens to identify factors that govern those phenotypes. Some of the things I didn't have time to talk about, but where we are also taking this in parallel is screening the non-coding genome. Everything I think I presented on here was about targeting promoters, but really annotating non-coding regulatory elements such as enhancers and silencers and insulators is where I think a lot of the strongest power of these tools is going to be. And then we have other studies on kind of expanding the CRISPR toolbox that I didn't have time to talk about, but hopefully these transgenic mice to take some of these concepts and move them in vivo will be useful. Hopefully I highlighted the students along the way that did the work. All the neuronal reprogramming was done by led by Josh Black, the satellite cell reprogramming led by Jennifer Kwan, and the transgenic mice were generated by Matt Gemberling. I, of course, want to acknowledge our collaborators that were critical to this work and the funding sources, but I'll go ahead and stop here in hopes that there's time for some questions. I'll also mention quickly that we recently were awarded our first NIH Center of Excellence in Genome Sciences Award, and this past fall, and it's specifically to develop tools and technologies for studying combinatorial gene regulation and annotating the function of the non-coding genome. And we've got several positions posted associated with that. And so if this is an area of interest to you, please feel free to reach out. And thank you again for the opportunity to talk about the work.
Thanks, Charlie. That is impressive as usual. I don't see any questions in the Q&A box. I think people are just too much in out. I just want to say that if you have questions for Charlie later, you can always put it in the box, and Candace can send it over to Charlie for him to answer. I do have a couple questions for you. So when you use activation to differentiate iPS cells into different lineage, how transient can that be? Do you have to deliver it like with plasmid or on virus? Have you ever tried to just use RNPs or messenger RNAs to do the activation?
Yeah, it's a really good question and something we spend a lot of time thinking about. I think it's hard to come up with a good answer. I think that to your specific question, I have seen, there is work using like RNA to get RNA encode, to encode the epi editors on RNA and, deliver the RNA to get changes in gene expression. I don't believe that I have seen that done with RNPs. And so that may imply, and we've tried that, but I don't think we've tried very hard. So it's possible that that implies that an RNP is too short-lived to get the effect, but that RNA might be enough. I think that what we know from the whole body of work of everyone in this field is that the effects on gene expression and epigenetic state are very highly dependent on the baseline state of the target and the gene, the duration of expression, the level of expression, which editor you're giving, the guide RNA, like every detail you can think of seems to affect this. And so, I don't think there is going to be like one simple answer of, oh, you only need it for 2 days or 4 days. It's going to depend, I think, on all of the other parameters of, how strong of a change are you getting, which epigenetic marks are you changing, what's the baseline state, is there, one of the things we take advantage of in the examples that I showed is that there's kind of positive feedback built in. Like once you turn on a neuronal gene network, it wants to keep itself on. It's fallen into one of those valleys in the Waddington landscape, and but that's not necessarily the case for all genes. So it's complicated.
But for therapeutic purposes, it's probably better if you use something shorter lived to reprogram them and inject in vivo, right?
Sorry, you said that it's therapeutically it's better if it's shorter lived.
Right. No, for like shorter lived for those factors you put in, yeah. But they still have to have a stable effect.
That's right. Yeah. So, I mean, I think an ideal, epigenome editing therapy would be that you can transiently deliver the therapy, you can have it go away, and you can have a sustained effect.
Right.
I think that it's interesting to think about, is that essential in all cases, and kind of what are all the parameters that govern that? But it's a good point.
Back to at the beginning, you talked about dCas9 seems to have no problem accessing closed chromatin for binding. Do you think that applies to gene editing, so you don't have to worry about chromatin structure at all?
Yeah.
And so timing-wise, maybe, what about HDR?
Yeah, I may have overstated it, but I don't want to say, hopefully I didn't say that you don't have to worry about it at all or that it doesn't matter at all. I think that there is strong evidence that if you average the activity of Cas9 or dCas9 for editing or gene editing or epigenome editing, if you average that over hundreds or thousands of sites, in general, you're going to have stronger activity at open chromatin than at closed chromatin. And several groups have shown that pretty conclusively. I think my pet peeve is I think some people have overinterpreted that to say, oh, you can't target closed chromatin. And I think that there's quite a bit of literature out there indicating that you can, both for gene editing and for epigenome editing. All things considered, if you have the option, target open chromatin, I think, but I don't think you have to restrict your experiment that way.
My one last question is on the CRISPR-I screening. You use multiple guide RNAs for each promoter region to turn on the gene expression. And in your screen, screen up the positively selected guide RNAs, how often do you see all 5 designed to one gene in your enriched pool?
I'm sorry, can you say the question?
How often do you see all 5 of the guide RNAs designed to one promoter region to be enriched in the pool?
I think never. We've never, and there's a couple of issues here, and I think people like John Dench are probably much more equipped to speak to this than I am. But I think a couple of things. One is that I think at most, we saw 2, maybe 3 enriched, right, of the 5. However, there's a lot of details there, right? I mean, if we had, maybe sequenced deeper or had more samples, maybe other ones would have popped up. Also, what was I going to say? Those were libraries that were designed, pre-2015, and I think that there's I think the library design has improved since then, and we pay very close attention to people like John Dench's work who are continuously improving those libraries so that we can get a higher hit rate and not have to use as many guides. What we did notice was that when we got hits, when we looked at the other guides for that same promoter that weren't hits, they tended to trend in the right direction. So one of the things we also know is, I mean, the statistical cutoff that we're using for calling a hit is arbitrary, right? And so, I think if we play with that, we can change some of those numbers, but they all would trend they would tend to trend in the right direction if there were one or two hits.
Thanks. If there are no more questions for Charlie, let's thank Charlie again. We will move on to our next talk. Our next speaker is Abraham Kurt. He is a graduate student in Keith Jones' lab at Harvard, and he will be talking to us about the exciting first transversion base editors. His title is CRISPR C2G base editors for inducing targeted DNA transversions in human cells. Abraham, please.
Hi everyone. Thank you very much for the kind introduction, Shasha. I would like to first start by thanking the organizers, Shasha, Candace, Jonathan, and the rest of the organizers for organizing this event and inviting me to give my talk about our recent work in our lab. Let me share my screen. Right. So today I'll be mainly talking about our recent work in the lab, describing C2G base editors that are functional in human cells efficiently. Obviously, it's a teamwork, and specifically, I co-first authored this work along with Rango Chow in Dr. Keith Jung's lab, and our lab is mainly genome editing and epigenome editing tech dev lab. And a quick disclosure, I am a co-inventor on a patent that describes our work that I'll be talking about today, namely CRISPR C2G base editors, in short, CGBE. All right, so because that's the day one of the conference, and I'm the second skip speaker, I thought I would give some background on the genome editing tools and shift some gears from Charlie's great talk of epigenome regulation into genome editing tools. So the field really has been rapidly developing, probably like started 3 decades or so, by the meganucleases. There are large proteins found in bacteria and yeast, mitochondria specifically. There are really large proteins, 18 base pair cutters that can bind and cut specifically, but they proved to be quite hard to engineer for a common lab across the world. Then the advent of zinc finger nucleases was quite exciting for the field, mainly because zinc fingers were naturally found in the human genome, and that was thought to be a good point because you want to avoid immunogenicity problems. However, it turned out to be not as modular as people thought to be at the beginning. It turned out each domain somewhat affects the flanking domain. So that also proved to be quite hard and above the threshold that's needed for a common lab to design and use this genome editing tool. However, what I would call probably the most modular one, transcription activated like effector domains fused to FUC1 nuclease, talons in short, was another excitement in the field. However, due to few amino acid changes between each domain, translating into few base changes in the DNA, proved it hard to also design and clone as side by side due to repetitive regions. However, as we all like to say, CRISPR-Cas9 democratized in the field genome editing tools. And mainly, I believe the reason is, right, so the first 3 tools that I've spoken so far, namely meganucleases, DFN, and talons, mediate the interaction with the target DNA via protein DNA interaction. However, CRISPR-Cas9 does this, as we all know, by RNA DNA Watson-Crick-based pairing interaction. That's a big difference and makes the cloning quite easy and the feasibility increase dramatically. Long story short, all of these first versions, at least, were aimed to induce double-stranded break at DNA. And as Charlie beautifully presented and alluded to, you don't really always want to introduce double-stranded break. That's not always the best and most interesting outcome that you want to achieve, given the preprint about the chromatopsis potential activity of Cas9 that we might talk later in the panel, perhaps. All right, so as we all know, again, the double-stranded break, mainly in mammalian cells, can be repaired by two paths. The first one is called non-homology and joining NAGJ, in short. That is somewhat unpredictable, although there's a great deal of literature now also suggesting microhomology is the main driving force, but nonetheless, unpredictable leads to insertions and deletions and gene disruptions. Or in the presence of sister chromatin or an exogenously provided template, homology-directed repair can take place to resolve the break. However, the components that are required for HDR pathway mainly effectively expressed at cell stages of S or G2 before mitosis. And that's why that really limits this application to really mitotic cells and virtually probably almost impossible in non-dividing cells. And the efficiencies are quite low as opposed to NHEJ, which is active all the time in all cell stages. So that's the reason I'm talking about this, is to mean that the precise DNA editing, and in this talk, when I say precise, it's not the accuracy, meaning off target, but the mutation that we want to install, how effectively we can install it without inducing of byproducts. However, the advent of base editors and the prime editors, next generation, I would call genome editing tools by the amazing work of David Liu and his team, really dramatically increased the precision capacity. And mainly, I would say this is possible because base editors and prime editors are not really intended or aiming to introduce a double-stranded break. Rather, they induce a single nick by theory to induce more precise alterations as opposed to nuclease Cas9. So I'll switch more into base editor and come back to prime editor towards the end of my talk. All right, so at the time when we published this paper, base editors came in two distinct flavors, either CVE, namely cytosine base editor, that can introduce transition C to T pyrimidine mutations, or ABE, A base editor, that can do a purine A to G changes. And here specifically, we're seeing schematic of a CVE that's comprised of an S and nickase Cas9 fused to deaminase, fused to uracil glycosylase inhibitor. So upon binding of Cas9 to its target, nicking Cas9 to its target, that leads to generation of what's called R loop or bubble that gives a partial access to deaminase to potentially deaminate its target cytosines in the editing window into uracil. And UGI is believed to be protecting this uracil from base excision repair pathway, mainly by blocking the endogenous cellular machinery component called uracil and glycosylase that will try to come and excise this base. That eventually leads to C2P. So in summary, base editors, without the need of a precise, without the need of a template, can install precise repairs, can be quite efficiently. It can be up to 80–90% depending on the site, and without really inducing much indels, virtually no to very low indels compared to conventional Cas9. However, CBEs and ABEs cover only part of the mutations that one would like to install in any given organism, in any given genome, especially in human cells, that is summarized in this pie chart. And this pie chart is showing the changes that are needed to reverse human pathogenic point mutations and that are at least cataloged. And the most relevant part of this pie chart for our talk is this 11% light blue on the left that is needed as C to G changes to reverse certain human pathogenic synapse. So that's why the field was in dire need for other types of base editors. Our study project really kicked off by this unexpected observation that also some other groups, for example, Jinsu Kim, also observed and published, came with the observation that ABE, that is intended to deaminate A, can also induce C2G edits at protospacer position 6 specifically. So here we're seeing data from 3 protospacers. Each box is one guide, hex site 2, AB site 7, hex site 1. Each row is one replicate of one sample treated either with control or AB max or one of the secure max variant that is described by our lab, by Julian Grunwald in 2019 with the rest of the team. So secure variants essentially have reduced RNA off-target spurious editing activities. The main message here is, if you look at these heat maps and the coloring in the heat map correlates with the C2G editing activity, we see at all sites across the board, we see varying levels, but consistent editing at the C, 6 into G. So that led us to hypothesize that perhaps the E. coli tRNA specific adenosine deaminase TADA found in the ABE structure can perhaps also deaminate the cytosine into uracil, as well as the adenine into inosine in the editing window as shown here. So to test this hypothesis, we first decided to fuse 2 uracil glycosylase inhibitor UGIs, twice of these domains to the C terminals of the AB max, similar to the B4 max structure. So here I'll be showing data, and across the presentation, I'll be showing data mainly from HEC293T cells. Towards the end, I'll show from K562 HeLa U2S as well, but mainly from HEC cells. And mainly the data will be targeted amplicon deep sequencing of different outcomes of population of cells. So here, each column is one guide, and we are focused on the 5 bases with the C6 centered in the middle in the editing window for each guide. And each bar is a replicate, and the outcomes are shown as in the legend. So for example, yellow would be a G, and red will be a T, and blue is a C. So here we're seeing specifically zoomed into this inset, RNF2 site 1 and FANCAP site 1. When we compared AB max side by side with AB max with 2G UGI, we observed that C2G editing activity almost completely abolished and replaced by C2T, and the indels, that few indels at the top shown by gray, were gone. So that strongly suggested, if not proven, that our hypothesis was correct, that ABE can probably deaminate the cytosine at position 6. So building upon this observation, we figured that it's better to start with, if we want to get an efficient CGBE, it's better to start with an deaminase like rat APOPAC1 in CBE constructs. It's better to start with one of these enzymes that is intended in the first place to deaminate a cytosine. And we figured that if a basic site generation by excision by the endogenous UNG is required, we figured that we would try to first remove the UGI and get CB delta UGI and fuse either human or E. coli UNG further by rational protein designing. So I'll start showing some data. So that's the same 7 sites from the HEC cells, showing B4 max versus B4 max delta UGI without the UGIs. All right, so first thing, as expected, we observed, as shown by this gray top part across all boxes, we observed high indel frequencies. And that was expected because you are effectively creating double-strand break more efficiently, because you have one nick on one strand now, and you are allowing the endogenous UNG to come and generate an A-basic site by excising the uracil that was deaminated. But interestingly, we started seeing C2G editing popping up, especially in the RNF2 site 1, 32%, up to 32%. It varied across sites. Sometimes it wasn't there at all. Then when we, then side by side, when we were doing the experiment, we also tested the human UNG fusion, as I said. And we tried different conformations, but because of time and space limitations, I will not be showing all the data. We observed that the human UNG fusion didn't really optimize the outcome. If anything, it actually reduced the C2G editing activity. So we decided to try the E. coli UNG as well. side by side. But before that, because we didn't know the exact mechanism why a C is preferably altered into a G, we didn't know the exact mechanism. We thought perhaps the deaminase, the dynamics of the deaminase playing with it by installing different mutations could increase the efficiency of CTG editing. To this end, we decided to try R33a mutation that was also described by our lab earlier by, again, Julian Grunwald et al., where they described the secure CBE variants. R33a essentially also leads to dramatically lower off-target RNA editing activities. And when we installed this mutation into the same construct, we were happy to see that CTG editing activity was higher, at least in RNA site 1. Some sites, it didn't change much. Some sites, yeah, not even, maybe lower, even 14%, 11%. It was definitely context dependent. Then side by side, when we tried the E. coli UNG instead of human UNG, and we tried on both conformations, N-terminus, C-terminus, specifically the N-terminus conformation proved to be the highest efficiency. Here we're seeing RNF2 site 1, for example, 63% activity, kind of quite pure product. And compared to B4max delta UGI, it's almost doubling. And another maybe extreme example would be FANCAF site 1, while there were almost virtually no CTG editing activity happening here. Now we were seeing 23%. Some sites, again, it varied. It didn't affect much. So definitely it was site specific. Then we tested our CGB construct, which we named as CGBE1. I forgot to mention here. So we test our CGB1 at more sites, and actually in the paper, total of 48 different sites we tested, but I won't be, of course, showing all of them. Here are some example sites, side by side compared with R33a, B4max. We were happy to see, while it was still context dependent, we could get as high as 71%, for example, here, quite pure product purity, Hex site 2 site, AB site 8, 52%, AB site 7, 62%, quite pure, as well as really low 9% with low product purity. And when we looked closer, we realized usually when high product purity and high efficiency was achieved, the C6 was flanked by either an A or a T, or W as a UPAC nomenclature. So that was an interesting observation back then. Then, because we never tried actually trimming the E. coli UNG away, we thought, what's the impact of getting the E. coli UNG away in the R33a architecture? Because that would be obviously very useful. Smaller is usually always better in the genome editing field, obviously for therapeutic purposes, AAV, packaging limitations, so on and so forth. So when we tested this and named it mini CGB1 side by side, we realized, and I was personally at least a bit surprised, that there were really like modest decreases, like for example, from 66 to 61% here, and then 49 to 48, 68 to 67. There was like modest decrease in the indel frequencies, also some modest decrease to the indel generation as well, which makes sense when you remove the overexpression of a UNG. So when we looked at the data aggregated from all the 48 sites, it turned out that CGB1 and mini CGB1 both had a favorable editing window at base position 5 and 7, with the 6 being the most favorable. Then we also wanted to check the Cas9 dependent off-target profiles of our CGB1 constructs, comparing to B4max and nicking Cas9 side by side as a negative control. And to this end, we chose few sites with a cytosine 6 in the middle at position 6. I apologize. And we focused on the top potential off-target sites as identified by GuideSeq. Actually, Shangdar, they will give a talk to us today, hopefully. Did publish in our lab. That's essentially an unbiased way of genome-wide identification of off-target sites for nuclease Cas9. So we focused on the top sites from this data set. When we looked at it, So here we're seeing the B4max on left panel. We're seeing the on-target activity on C4 and C6 around 50%, and here we are seeing CGB1 up to 82% on the C6 only. And when we look at the top of target for the hex site 2, we were able to detect 1% or 2% activity happening for B4max, whereas it wasn't really detectable for CGB1. Another example for Cas9 dependent off targets, hex site 4 this time. Across the board, although this time the on-target efficiency of CGB1 was also lower than B4max, so that's why it's hard to really correlate why the off-target efficiency, off-target frequency of CGB1 is lower, but nonetheless across the board, it looked more favorable to B4max side-by-side comparison. All right, so then concerned about now the extra parameters that makes it more strict for targeting range, right? Like now we have the AT enrichment flanking the C6, which needs to be experimented further though. Then we have the PAM, and we have the cytosine at position 6. So all of these compound that we were worried that limits the targeting range more than other base editors. We wanted to test how about the relaxed PAM variants of SPCas9 or altered variants of SPCas9. We tested Cas9NG variant that can recognize NG shorter PAM as opposed to NGG. And we tested on a few sites, like 6 sites. But we were happy to see, although we need to do more experiments, but we were happy to see that at least they are both CGB1NG and mini CGB1NG architectures were functional. And when we tested Cas9 VRQR, that was also defined by our lab, by Ben Kleinsteiber, that can recognize NGA PAMs as opposed to NGG altered PAM specificities. We were also happy to see activities of CGB1 VRQR and mini CGB1 VRQR at different sites at different levels. So that was encouraging to see that altered or laxed PAM variants can, to a certain extent, accommodate this more strict parameters and expand the targeting range of CGB constructs. Okay, so finally, while we're wrapping up our project, the amazing tool by Anselm et al. And David Lu's lab called Prime Editing came out, and we wanted to test side by side with our CGB constructs. So one quick disclaimer is this was really an out-of-box direct application as opposed to comprehensive scanning of all potential PEG RNAs and nicking guide RNAs. We picked the 4, but nonetheless we followed strictly all the recommendations that were outlined by Andrew et al. In the paper in terms of RTT length, PBS length, nicking guide RNA distance in the application of PU3. We followed strictly their recommendations. We tested in 4 different cell lines: HECT cells, K562U2S, HeLa cells, each of them in different boxes. Each group of columns are 1 site, and these are sites that we already knew that we could induce high levels of CDG mutations in human cells. And when we did an out-of-box application side by side, we observed few things. First of all, we were able to reproduce in accordance with their findings that PE3 always outperformed PE2, and that PE3 was the most active in the HEX cells, again in parallel with their findings, and that CGB1 outperformed across these 4 sites at least prime editing, which is again in parallel with their findings when they test base editing. They also came to the conclusion that if you have a good target site where you have a PAM and a good editing window for your region of interest, it's better to use base editor. You can achieve higher efficiencies as opposed to prime editor that looks like one base change is not as robust as base editing. Okay, so some summary. We believe that CGB1 expands the research and therapeutic applications of base editing, as shown by the box here. We believe this is mainly by unlocking new codon amino acid alterations that were impossible before. Here on left is the target amino acid, and then the bottom product amino acid that you can alter into. And the reds are editable by CB or AB, and blues are the new ones that are unlocked by CGB1 applications. And that's mainly because the wobble position, the third position in the codons, when you use CB or AB that can induce translation mutations, you heavily you lead to the same amino acid that's also visible by this diagonal effect in the box. Whereas when you use a transversion-based editor, especially in the wobble position, most of the time you end up having a new amino acid. And we also believe transversion mutations in non-coding part of the genome, especially in transcription factor binding sites, could have more pronounced effects as reflected by the literature. Sorry. So in conclusion, CGB1 can install CTG edits in multiple cell lines as high as 70%, especially in the AT-rich sequence context. SPCas9 PAM recognition variants are functional NG and NJ PAM, thereby expanding the range of targetable sites. CGB1 overall displayed favorable Cas9 dependent DNA off-target profiles compared to BO4MAX, and that CGB1 can potentially induce more efficient CTG edits compared to prime editing technology, again, in parallel with the original paper. And CGB1 unlocks new codon and amino acid alterations that were not possible with the current base editing tools. With that, I would like to thank, first and foremost, to my PI, Dr. Keith Zhang. He's been an amazing mentor throughout my PhD studies, and Rango Chow, the course author, Julian Grunwald, who co-supervised the work, Brett Miller, Lucas Langner, Samia Iyer, Sarah Garcia, Stacey Francis, and the rest of the lab. And finally, we are, I forgot to mention at the beginning, we are located at the MGH and at the same time, Harvard Medical School. And thank you very much. With that, I'll be happy to take any questions if you guys have. I know the time is a problem, but I guess I started a bit late as well.
No, you're good. Thank you, Abraham. I do have an announcement to make first. I just realized why I was the only one who's asking questions to Charlie before. It turns out the panel, all the speakers don't have access to the Q&A box. So if you have a question, just turn on your microphone and ask. So anyone has questions for Abraham? I'll start with one. Your lab is obviously very well known for looking into off-targeting events. So have you putting any effort to look for the CRISPR independent off-targeting for these CGBs?
Yeah, we haven't, that's a very good question, and we haven't done this yet, but it's definitely on to-do list. Yeah, we haven't done this specifically for CGB.
Expect it'll be kind of similar to the ABs and
yeah, it's a good question. Like thinking that so first of all, we know right from the 2019 paper that I alluded to, R30, so CBE, the canonical CBE with rat APOPEP1 has potential to induce both RNA and DNA, spurious DNA off-target DNA RNA editing activities. But then we also know R33a dramatically reduces this, although not completely abolishes this. So because we have R33a in CGB1 and in CGB1, I would expect, again, needs to be shown empirically, but I'm speculating here, I would expect it to be lower, similar to its parental R33a.
Oh.
I have a quick question, if I may. This is Shendar Ibrahim. Nice to see you.
Hi, nice to see you.
I was definitely interested in the results that you had with the mini CGBE versus the CGBE, and I'm wondering if you can kind of speculate on the mechanism that you think of C2G conversions based on kind of your recent data.
Yeah, it's also shocking and amazing to me as well. So first of all, when you compound this finding with the other recent papers, one of them published with us back to back, the Chow et al. And one by archive paper, Chen and Chu et al., this Singaporean group, right, where they also did similar stuff. To me, it looks like, again, all of these needs to be empirically tested, and I'm just speculating at this point. But first of all, it looks like you don't really need an exogenous UNG. Perhaps the internal endogenous UNG is enough. But at the same time, there's this interesting concept, why R33a, like we don't really know the mechanism, but why it really increases the efficiency of CDG editing. That's, I think, the real answer lies somewhere there. But what's even more interesting, if you look at the literature of DNA repair, is in the cancer signature hotspot papers, where they check the endogenous human R33a activities. They also see similar A and T flanking a potential C becoming a G. So I believe, so again, speculating, I believe somehow what's called translation polymerases are playing a role here. When the A basic site is generated from uracil, either it looks like there are two potential ways. Either this A basic site is directly replaced by a C, by a translation polymerase like kappa or beta, or the nick induces a QBAG, and on the opposite strand, like Rev1, can also, when you have an A basic site, can install a wrong G opposite to that. So I believe the answer lies somewhere around this realm, but more detailed experiments needs to be done.
Okay.
So I noticed some of the, so for most of the sites, you have different base changes, not only to G. Among all these target sites you tried in different cell lines, did you try to single cell clone just to see, are you able to get homozygous change at a site, or mostly it's going to be mixed, or one allele is unchanged?
Yeah, that'll be very interesting to do. Unfortunately, we haven't done any single cell cloning yet, but if we ever do, I'll be happy to pass you the information, but we haven't.
All right. If there are no more questions for Abraham, we will go to our next talk. Thank you, Abraham.
Thank you very
much. Our next speaker is Trevor Hart. He's currently an assistant professor at the Department of Bioinformatics and Computational Biology at MD Anderson. And he got his education in the very fine school of UT Austin. It looks like we overlapped for quite a few years. Trevor's talk is CRISPR screening of functional genomics and cancer targeting. Trevor?
Thank you. Thank you for the introduction. And I agree, UT is fantastic. I went there for my undergrad and my graduate work, and would happily go back as a PI if they gave me the chance. Austin's one of my favorite cities.
And Middle Eastern studies for
yeah. So my background is a little bit nonlinear compared to, I think, most of my peers here. But yeah, so my undergrad is in Middle Eastern studies, and my PhD is in cell and molecular biology, which makes perfect sense. But here I'm going to talk a little bit about CRISPR screening for functional genomics and cancer targeting as a continuation of my postdoc work in Jason Moffat's lab at the University of Toronto, and then ongoing work here as assistant professor here at MD Anderson. And I'm going to talk, well, first of all, disclosures. I am a consultant for repair, but none of that's related to anything I'm talking about here. So the three things I'm going to sort of touch on are what is a hit in one of these screens, and I'm going to approach this from an opposite perspective of the first two speakers, and we're going from the big picture of integrative analysis of a large amount of data down to individual sort of functional genomics approaches. So what is a hit? Why is a hit in a particular background? And then the sort of exciting story from my perspective is what are we missing? What are these genetic screens not able to detect? And so I think everybody here is familiar with the construction of the anatomy of a whole genome screen, but it's worth reiterating and amplifying a couple of points. So to generate your CRISPR library, you generally order oligos off of an array, and you clone them into some viral vector, and you package them, and you transduce them into cells at a low MOI such that each guide is represented in a single, each cell only uptakes a single guide. So there's a single genetic mutation per cell in a pooled screen. But then each guide is represented in somewhere between 100 and 1,000 cells, so you could get good, robust ability to detect phenotype from that individual guide activity. And then you take some reference distribution of the relative frequency of each guide in the population at the beginning and at the end of the screen. And the end of the screen is somewhere between 14 and 21 days, or on the order of 10 doublings. And so what that really means is you're looking at, for a typical genome scale library of about 70,000 guides, you're looking at 30 to 50 million cells per replicate that need to be grown at least 10 doublings. And that means cell lines. That means it's hard to do this in primary cells or anything that doesn't allow you to grow cells in that sort of scale over that sort of time. And so that's what all the work I'll talk about today is. It's all done in cancer cell lines. So how do we do analysis of screens? So I mentioned that we're looking at relative abundances of individual guides. We have a piece of software called Bagel that looks at the relative fold change of a set of known essential or non-essential genes. A good screen looks like this, where the guides targeting a non-essential gene have a fold change that's roughly normally distributed around 0, and then the essential genes drop out relative to that. We have an updated piece of software called Bagel 2 that's been in use for a couple of years now, but we finally got around to writing the paper, and that's out now. But I won't go into that in any detail here. I'm happy to talk about that offline. The real quality of Bagel was shown in the early days of screening when the data wasn't that good. Now that the data generally is as good as what's shown in this left panel here, the methods don't matter as much. The signal to noise in the data itself is quite high. And so most of the different methods give you basically the same results. But what's important to consider here is that when we talk about what is an essential gene or non-essential gene, we are talking not just about the extreme phenotype of you knock out the gene and the cell dies, but we also see just a continuum of fitness defects. So when you lose a gene, that cell slows down, has lower fitness, doubles at a slower rate, and so that cell gets outcompeted by other genes that don't have fitness defects, and so that results in a dropout from the population as well. And we call these essential genes, but it's not necessarily something that causes cell death. It's something that causes some moderate to severe decrease in fitness over the course of the screen. So even in the early days, and this is our sort of first or second generation screen, even in the early days, we were able to determine these very highly specific context-dependent gene essentialities. So this is the nice case that we showed in our first paper. This is from Jason Moffat's lab, where depending on the background state of P53, you could determine whether or not MDM2 was essential or not. You could very clearly predict the MDM2. So MDM2 is the native regulator of wild type P53. The first thing any tumor cell does is shut down the proapoptotic function of P53 in a P53 wild type background that is almost always by MDM2, which is highly essential in those backgrounds. But in the cell lines that we screened, or in the case of A375, the previous data that we reanalyzed, we show that in DLD1, which has a P53 oncogenic mutation, the MDM2, MDM4 pathway is not essential at all. In HeLa cells, which are regulated by an entirely different pathway through HPV encoded E6 protein, leverages endogenous UBE3A to circumvent the MDM2 dependency. MDM2 is non-essential, and UBE3A is specifically essential in that background. So that really, I think, launched or helped launch the field of background-specific vulnerability assessment to try to learn specific cancer targets in specific backgrounds. And so that data that is available now is quite extensive. And of course, John and company at the Broad have been the major drivers of this, with more than 800 cell line screens available now as part of the cancer dependency map, and then Project SCORE at the Sanger Center, or formerly Sanger Center, with another 300 plus cell lines. So this is fantastic, and that you can basically take your hypothesis of choice and see where or what genes might be associated, preferentially essential in those backgrounds. And so there's some obvious cases here. So here's KRAS, If KRAS is mutated, generally an oncogenic mutation, then KRAS gets a high base factor. That means it's highly essential, where when it's not mutated, it's non-essential. So this is fairly straightforward hypothesis-driven analysis. And this was done in a beautiful way in the Behan paper from 2019, the Sanger data, which showed the association between, in particular between oncogenic mutations and the likelihood that that gene is specifically essential in those backgrounds. So this is a really nice study. But that's sort of hypothesis-driven research. We take more of a data-driven approach to this to see what we can learn directly from the data without being sort of bound by frankly, what is my limited creativity in terms of what hypotheses to apply to this data. And so if you have some large number of diverse cell lines with different genetic contexts and different lineages, what we inferred from prior study in yeast was that if you had genes that were involved in the same biological process, which we call co-functional, then they were likely to have correlated gene fitness, knockout fitness profiles across this panel of cell lines. So this is a known thing from yeast, and to carry forward into the human cells didn't seem like any great leap, but it turns out to be strikingly true. And so our thought was, if we could find these modules of co-functional genes, then maybe we could identify the discrete sets of backgrounds in which they were essential and try to learn some causal basis then for why that module of genes was essential in that set of cells. And we could learn potentially cancer vulnerabilities in blue and cancer biomarkers or contexts in red at the same time, and learn it directly from the data. So we built this big correlation network and looked for modules in this network, and the modularity of the network is really quite striking. One of the modules that we find here that's centered around this protein called GPX4 is a glutathione peroxidase that's involved in inhibiting ferroptosis, which is a regulated form of cell death in cells. It's based on lipid peroxidation and shutting down runaway lipid peroxidation. Well, co-essential with GPX4 are these other 4 genes that just happen to be involved in the selenocysteine biosynthesis pathway. GPX4 is a selenoprotein, and so it requires insertion of selenocysteine at the appropriate locus. And so one of the key lessons here is just sort of what you would expect from a quality genetic screen is that whenever an enzyme is required in a particular background, then the cellular machinery involved in biogenesis and maintenance of that enzyme is also required in that background. What this doesn't tell us is what are the backgrounds where that's essential and why specifically. And so narrowing down into the data itself, we look at a panel of 20 or so glioblastoma cell lines that were in the initial data release. And we see that this pathway, or this module, is clearly essential in about 2/3 of these backgrounds, and completely non-essential in another set of, in a subset of this data. And we really don't have any idea why. So you start looking for other molecular markers of why that might be true. There's one strong association with epithelial versus non-epithelial or EMT phenotype. And the ZEB1 is a marker of EMT or non-epithelial phenotype, and where ZEB1 is expressed, the median base factor of this complex or this module is significantly higher, suggesting that this module particularly operates in non-epithelial cells. There's also a strong association with GPX2, another glutathione peroxidase gene, and its association with GPX2 is such that where GPX2 is not expressed, the GPX4 module is highly essential. And so that might suggest some level of synthetic lethality there, but there's a little bit of a confound because GPX2 is only expressed in epithelial cells. So which came first, the chicken or the egg here? We're not quite sure. In a very nice study that compared, this is the Lee et al. Paper from 19, that looked at metabolomic profiling of the CCLE cell lines in which these same genetic screens were done. It was this module, GPX4, was shown to be associated with cell lines that had high abundance of long-chain polyunsaturated fatty acids, which are the species that's associated with ferroptosis. And so there's a very nice biomarker that predicts GPX4 essentiality, but the underlying genetic causality of increased polyunsaturated fatty acid and GPX4 essentiality is still really not known. So there's a nice association, but no causality yet that we understand. And so this is one case of a module from a much larger network that we built. It's about 3,000 genes built across more than 400 at the time CRISPR knockout screens. And that really breaks apart into several hundred individual modules or clusters that each have their own associated set of backgrounds where they're emergently essential, and we don't know why. For about, like I say, there's over 100 of these. Maybe a third of them we can positively associate with either tissue or oncogene, and the rest, it's a fascinating and wide open research question. Beyond the simple modularity of this, we do see some hierarchy of the cell that's represented. And so we see that there's one strong module that's involved in peroxisomal beta oxidation that's highly coherent within these cell lines. There's an adjacent module that's involved in lipid homeostasis, which we've been particularly interested in lately. And then there's a related module involved in membrane trafficking, which seems to be associated on some level with the lipid and oxidative response pathways, but we don't have any hard experimental evidence for that. So this is a case where we would generate a hypothesis from the network that this RAB18 transport complex would be associated with lipid homeostasis in some way. And so that's one area of sort of unknowns, or letting the data be the guide to some of the biology questions that we ask. But one of the other sort of interesting questions that I alluded to earlier is, okay, we have this network, we have all this data. The network itself contains about 3,000 genes, but each one of these cell line screens targets 17,000 genes. Why are there only 3,000 genes in the network? What are we systematically missing from the CRISPR data, from the CRISPR whole genome screen data? And so here's one example that really highlights the problem. So this is the MAP kinase pathway, the RTK MAP kinase pathway, where we can see and then the individual gene essentiality scores across a subset of the cell lines at the bottom here. And what we clearly observe from looking at it this way is that the oncogenes that are essential in specific backgrounds are mutually exclusive. And so there's EGFR, and there's FGFR, and there's KRAS, and NRAS, and BRAF. And so those all define their own sort of discrete clusters of cell line essentials. That's all well and good, and that's to be expected. That's consistent with the tumor data. What we also see is these accessory proteins. So these proteins are essential whenever the upstream protein is essential, and so they don't show up directly on this map below, but it's pretty obvious that they are associated with sort of an either/or logic in terms of this network. And we can see, and we know that from structural and signaling studies. And so that we know why these particular genes are essential, because they are part of the protein-protein interactions and signal transduction pathways. But what is systematically missing is this last layer here, which is the MEK and ERK layers. And both of these layers are represented by gene paralogs that render this sort of step in the network as systematically invisible to monogenic perturbation screens. So if you knock out one gene, the other one takes over the load, and you don't see any phenotype in general. And this is particularly interesting in the case of trying to map drug response to these genetic perturbations. Because trametinib is just the classic kind of the canonical MEK inhibitor, there's almost no association between trametinib response, which is on the y-axis, and the gene essentiality profile of one of the genes that it ostensibly targets, MAP2K1, on the x-axis here. There's just no association there. But if you look at trametinib response versus the maximum base factor of the upstream genes at the RAS or RAF level, then there's a very strong response. So about 3/4 of the genes that show strong trametinib response, or 3/4 of the cell lines that show strong trametinib response, have a high essentiality score for one of these RAS or RAF oncogenes. And so, you know, based on the biology, we can make this association, but based on the direct mapping of genetic vulnerability and drug target, that connection is missing. And so that is, of course, because the paralogs are systematically missing from this data. And so what we did was we looked at gene expression levels across all of CCLE, and we defined 3 separate categories of constitutively expressed, never expressed, or sometimes expressed. And notably, between across these 3 groupings, in the constitutively expressed group, all of our genes that are sort of always essential are in that group, which is gratifying. And most of the context-dependent essentials are also in that group, which is also sort of interesting. But strikingly, almost half of the constitutively expressed genes across, you know, I guess at this point, more than 800 cell lines, are never essential in any of these knockouts. And if you look at if you compare these never essentials with the always essentials, and we look at the relative distribution of paralogs, and we sort of decrease the threshold of what is a paralog in terms of sequence identity, then we see that as we dial the threshold back from 100 to about 35% sequence identity, never essentials are more than 3 times more commonly represented among that set than common essentials, than these core essentials that are essential everywhere. So we know that never essentials are overrepresented for paralogs, which suggests that the paralogs are systematically missed by this data. And a computational analysis, or a couple of computational analyses actually show this to be generally true. And so this is our computational analysis where we take the set of paralogs and we look for loss of function of one of the paralogs to see if the other paralog shows increased vulnerability in cell lines. And we see on the order of 50 or 60 pairs that show very strong signal here. A nice study by DeKegel and Ryan out of Ireland shows a significantly larger number, and these don't overlap as well as you might hope, suggesting that there's actually a larger number out there that are either paralogs where the definition of loss of function is a bit weak, or where neither one of them shows loss of function, and those are of course systematically missing from all this data. And so we looked for a way to test this experimentally, and here we got into a nice collaboration with John Dench and used some of their data to generate a Cas12 screen. So we have transitioned almost entirely to the Cas12 versus Cas9. The advantage of Cas12 is that it will process a polycistronic guide array into multiple independent targeting units, which enables really sort of I would say trivial multiplexing, in that it imposes no additional burden beyond what a traditional screen does. So there's no mixing and matching of guides, which is great. It's been demonstrated that you can do that sort of certainly in the Cas9 background, but it's so much easier in the Cas12 background. The problem with the AS-Cas12 anyway, in our experience, is that it doesn't actually work. But certainly the enhanced Cas12 from the Jung lab in a Kleinsteiber publication works fantastically well, coupled with an improved guide design, working with Peter DeWert in John Dench's lab, and we were able to get this to work really well. We defined a very small screen targeting 2 genes at a time, and looking at differential log fold change of gene pairs versus the independent contributions for the single gene knockouts. The proof of concept here worked great. So this is single gene essential versus non-essential. So this is what the data was supposed to look like. This is great in one of our screens. And in the 3 cell lines that we screened, we get this nice long negative tail of negative fold change, negative differential log fold change, which represents synthetic lethality between paralog pairs. Of the 24 or so hits that we saw across the 3 screens at the z-score of less than -3, 14 of them were consistent across all 3 backgrounds, which is also very cool. And just walking briefly through some of the hits here. So here's MAPK1 and MAPK3, which is highly essential in all the synthetic lethality shows very strongly in all 3 of the backgrounds. HDAC1 and 2 also strongly synthetic lethal. So these are again paralogs, buffering paralogs that don't necessarily show individual gene knockout effects. And then some of the really interesting ones, I think this is a nice proof of concept here. So the COPS9 signalosome protein complex, the whole thing is essential everywhere except alternate subunits, COPS7A and 7B. Neither one of those is individually essential, but they show strong synthetic lethality in all 3 backgrounds. And then we were also able to show background-specific synthetic lethality. So this is cyclin E1 and E2 are strongly synthetic lethal, specifically in the OVCAR8 background, where CDK2, its cognate cyclin-dependent kinase, is strongly essential and strongly preferentially essential. So we're very happy with the ability to do multiplex genetic perturbation with Cas12, especially in particular with the enhanced AsCas12 platform. I'm very excited to continue to do work with that. It works as well as Cas9 in our hands. It makes combinatorial libraries very easy to synthesize and straightforward to screen. Analysis is non-trivial, but that's, you know, for someone from a computational background, that's certainly an opportunity more than a problem. And the additional sort of opportunity here is that the search space of potential gene pairs for synthetic lethals is enormously vast compared to the individual gene knockout space, which means that we have to do some computational prediction of what, you know, where the low-hanging fruit is before we go headlong into that analysis or into that screening effort. So to wrap up, I would say that integrative functional genomics makes it clear that this is a no-brainer, obviously, that cancer cells have been profoundly rewired. This is a known thing, but understanding the causal basis for the observed phenotypes that we see is not so obvious. We certainly see modular emergent essentiality in many different sets of backgrounds. We don't know why, and that's a fascinating question for us over and over and over again. And it's pretty clear that continued development, and apropos of this conference, continued development of screening technology is going to be required, both in terms of polygenic perturbation technology, but also gain and loss of function and the ability to do more precise mutations than complete loss of function as we've done in these screens so far. So with that, I would certainly like to thank my lab, John Dench's group, several other groups here at MD Anderson, and then my former collaborators at the University of Toronto. And with that, I would be happy to take any questions.
Thanks, Trevor. Any questions for Trevor? Go ahead.
Sorry. Hi, Ishan here. Really nice talk. So I'm just wondering, like, when you do, all the do the screen in all the cancer cell line, have you see like a unique pattern associated with the different cancer type? And that's the first question. And second question is like, for your GBM screening, because GBM has different subtype or grade, did you see any like module was consistently associated with a certain type of gray or class of GBM cell line?
Yeah, so GBM is a particularly difficult case because I actually work right down the hall from Jian Hu, who's a pretty well-entrenched GBM guy. His hypothesis is that all GBM cell lines are sufficiently plastic that many of the GBM subtypes are reflected in subsets of cells within that, even within that cell line. So the cell lines don't really do a good job of reflecting different GBM subtypes. There's a different argument to be made there about, you know, EGFR versus other, you know, particular dependencies, and Stéphane Auger had a nice paper a couple of years ago about GBM, the differences in GBM screens and GBM primary cells versus the GBM cell lines. In terms of the broader question of what are the lineage or oncogenic or genotype specific hits that we can find, absolutely, we do see those. And in fact, it makes it complicated in some cases to differentiate genetic from lineage specific effects, because, for example, the BRAF signature is so strongly associated with melanoma that it's difficult to detect BRAF outside of melanoma. But we certainly see some, so for example, melanoma specific transcription factors. And in fact, that's the most discrete, I guess, background specific hits tend to be the lineage specific transcription factors. Whether that's useful or not is an interesting question. Transcription factors are generally not considered druggable targets, and not necessarily viable ones either in most cases, because even if you could target it, you wouldn't want to, you know, knock out the entire tissue just to take out the tumor. So it's a different way of, a different lens of interpreting that is that there is enormous functional genomics information to be gleaned from this outside of the specific cancer targeting question, and that's really what we're trying to do, is, you know, learn the sort of functional hierarchy of any cell, not just a cancer cell.
There's a question from Rita Martinez. Great talk. With the Cas12a multiplexing, is there a limit to the number of CRISPR RNAs that can be multiplexed?
Opinions vary. We haven't really pushed it yet. That's on our very, very short list of things to do. Michael Reger and Tomas, whose last name I can't pronounce, from the Jason Moffitt lab, recently did a synthetic lethal screen called Chimera, where they used a single polycistronic array that started with a Cas9 guide and then had Cas12 guides afterwards. And they used LB-Cas12. And in their experience, you couldn't get more than about 4 guides to express properly. But I think others have shown that if you change the promoter to a Pol3 versus a Pol2 versus a Pol3 promoter, then maybe you get longer, more effective transcripts out of that, and maybe you can get longer effective Cas12 arrays. I'd be curious about John Dench's feedback on that if he's around.
Well, maybe we can.
Of course, you said that as I was getting up to get a glass of water. But yeah, we have done 3 guide arrays using only Cas12a, and that works very well. We haven't gone higher yet, but you know what I worry more about is the more guides you have. If you're using, you know, nuclease active Cas12a, you're just interested in lots and lots of cuts in your genome. So that might get to be more limiting sooner than the processivity of Cas12a for processing guides. But that's a guess.
Yeah, so that's a concern that we certainly have, but at least our paper argument against that, or our paper hypothesis for why that might not be true, is that the Gonsalves et al. Paper seems to indicate that high cytotoxicity from multiple cuts is because of locally amplified loci instead of multiple cuts throughout the genome. So maybe that's less of an effect than we think it will be.
Yeah, which would be great, obviously, but we just don't have the data yet to know.
Right.
I actually have a really naive question, not being in the field of screening. So is it, if say you're really looking for 2 genes being synthetically, so can you just do the library twice, introduce the library twice after one round of selection, then apply the library again so it can pick up another guide RNA? Would that work at all?
So there's 2 ways to think about that. One is that you introduce a single guide So you got a single gene knockout, and then introduce your second guide. That's certainly one way to do it. And John has better background on this. In fact, they published a paper on this entirely. So they've used the alternate Cas9 species to do different to combinatorial knockouts. And so you can do this sort of anchor gene approach, but it's trivial to do with the Cas12 because you just introduce, your one gene of interest and then your other set of genes that you want to test genetic interactions with. And so the real value, certainly the real value of the Cas12, I think, is that it's trivial to synthesize these libraries because all you have to do is synthesize your oligos the same way you would, at least for a dual guide library, you just synthesize your oligos the same way you would a Cas9 library. And there's no, generating a single knockout clone that's required in advance. You can do all the pairwise perturbations in the same pool.
I have a quick question. So great talk, Trevor. Is the transcleavage activity of the TPF1s, I know it's a hard question, but do you think it has any implications at all? Like I believe upon binding or editing the on-target, right, there's a transcleavage activity happening for single-strand DNA and double-strand DNA. Do you have any insight, like what's happening in the cell? Does it have any impact at all?
I'll be honest with you. Our readout is does the screen separate the essential genes from the non-essential genes at the end of the day? And anything that happens before that, it doesn't matter as long as those curves get separated. So the answer is no. We haven't looked at that at all.
I see another question. What is the state of polycistronic guide RNA library construction for use with EN Cas12? For example, transcription factor library, as mentioned earlier.
So we have not generated or published any libraries except our small scale Paralog library that we mentioned earlier. So that's published. The Broad, and in particular John's group, has done an amazing job of generating individual guide designs that we think are probably generally pretty accurate and active and specific, which is, those are equally important things. And they have systematically looked at different direct repeat sequences. So the CRISPR, the Cas12 scaffold region of the guide can be modified slightly which facilitates multiplexed arrays, basically. So you don't have to have too many repetitive sequences in your array. So the short answer is no, there are no polycistronic guide libraries available today, but the individual components are available.
Great. Thanks again, Trevor. We will move on to our next talk. Our next speaker is Shinder Tai. So he is currently an assistant member at St. Jude's Hospital. And he is probably best known for GuideSeq because so many people have been using it. And he was also the one who first published on the pair nickase of Cas9, but GuideSeq has been outshining over him. And since he left Keith John's lab and starting his own lab, he's published CircleSeq and ChangeSeq. It looks like every couple of years there's a new seq coming out of Shinder's work. Today his talk is understanding genetic and epigenetic effects on CRISPR-Cas9 genome-wide activity with ChangeSeq. Shinder?
Great, thanks Shasha for the kind introduction, and thanks to all the organizers for inviting me to speak. It's really a pleasure to be here. Today, I'm going to tell you a little bit about two stories today, I guess a short snapshot of some of our efforts on therapeutic genome editing, and then as our title implies, more understanding the genome-wide activity of genome editors with ChangeSeq. So some quick conflicts of interest disclosures. Okay, so I think our lab, is, many others are really excited by the promise of genome editing therapies. This includes potential monogenic disorders like Huntington's disease, cystic fibrosis, the change muscular dystrophy.
Shankar, I don't think your slides are being shared.
Thank you.
I mean, you look great, but I don't want to see your data.
Let's see. Slight oversight there. Okay. How's that?
That looks good. Thank you.
Okay, perfect. Thank you, John. Okay. So again, thank you. Conflict of interest disclosure. So yes, genome editing therapies. We're really excited about the promise of these genome editing therapies in terms of monogenic conditions, as I mentioned over here before that you couldn't see, but also others like cancer immunotherapy, and in particular at St. Jude at conditions like sickle cell disease and other hemoglobinopathies. I think we're particularly excited about this because of the potential for these to be curative genetic therapies, where you could treat patients one time with this particular therapy and cure them of their particular genetic condition. The therapeutic vision that we share for these autologous cell therapies might go something like this. For beta hemoglobinopathies, for example, we might isolate human hematopoietic stem cells. We'd mobilize them for patients, isolate CD34 positive HSCs. We might perform some type of productive genetic modification to them. We would condition the patients to be able to receive these stem cells, reinfuse them back into the patient. And so some of the advantages of these types of autologous cell therapies would be that there's no issue of donor cell availabilities. It eliminates the risk of graft versus host disease. Some of you might be pretty familiar with sickle cell disease, but for those of you that are not, here's kind of an overview. It's the first molecular disease to be characterized. It's caused by a genetic condition. It's caused by a point mutation in the HBV gene, and it's associated with severe pain, chronic anemia, progressive multi-organ failure, and really early mortality, where a lot of sickle cell patients really die in their 50s. There are pharmacologic treatments such as 4-hydroxyurea for sickle cell disease, but these drugs are not curative, so they can kind of treat some of the symptoms, but they don't really prevent the progressive multi-organ failure or the early mortality. One curative method to treat sickle cell disease would be bone marrow transplant. And so this might be with a matched sibling donor, for example. And so actually, it was when I came to St. Jude, I was surprised to find out that the actual first curative transplant for sickle cell disease happened here at St. Jude. One issue with it is that there are limited numbers of haploidentical donors that are available. So, there's not really a matched sibling donor available for everyone that does not have sickle cell disease. At St. Jude, we treat about 900 patients with sickle cell disease in our Department of Hematology. And so one approach that we've been really interested in is the idea of hemoglobin switching as a potential therapy for sickle cell disease. What's interesting and known is that there's a developmental switch from fetal to adult hemoglobin that happens around the time of birth. And what's really fascinating is that there are people walking around that have sickle cell disease mutation, and they also have another mutation that causes hereditary persistence of fetal hemoglobin. And people that have both of these mutations are actually totally asymptomatic typically. And so it's been thought for quite a long time that induction of fetal hemoglobin is a potential replacement for adult beta globin. And so our approach for trying to treat sickle cell disease is the idea of trying to induce fetal hemoglobin as a replacement for the defective sickle adult beta globin. One of the rationales for this kind of approach is that you might think it's easier to simply just directly correct sickle cell mutation, and that is one of the long-term goals. But with current genome editing technologies, non-homologous end joining is really much more efficient than homology-directed repair. And so it's simpler to try to disrupt some sort of genetic element to induce fetal hemoglobin rather than directly correcting sickle cell mutation. In addition, induction of fetal hemoglobin might be more generally applicable to other beta hemoglobinopathies, including ones that are caused by defective transcription of beta globin. And so here's the editing target that we're really interested in. It's in the fetal hemoglobin promoter. We'd like to edit this repressor binding site of a repressor called BCL11A at the human gamma globin promoter to try to induce fetal hemoglobin as a replacement for sickle beta globin. And this is kind of the schema of autologous genome edited cell therapies that we envision. So to try to understand whether this is working, this is kind of the experimental design for editing of human hematopoietic stem cells, where we're transplanting them into immune-compromised mice. And so the experiment is really designed to analyze genome editing of repopulating human hematopoietic stem cells 16 weeks after transplantation. It's important to do this because otherwise, when we're looking at bulk human CD34 cells, there's kind of a population of both stem and progenitor cells. And so it's not clear that you're really editing the true stem cell population. So here we're mobilizing human hematopoietic stem cells from sickle cell disease patients using an agent called plerixafor. We're isolating these human CD34 cells from the patients. We're stimulating them with cytokines, electroporating them with Cas9 ribonucleoprotein complexes, and then we're transplanting them into these NBSGW immune deficient mice. And so on this next slide, I'm really showing you a summary of about 2 years of work in trying to optimize this process. One aspect that was really helpful was a collaboration with Scott Wolf and Dan Bauer's lab, where we're using a version of Cas9 that has a stronger nuclear localization signal. And we're finding that these end up being able to better edit human hematopoietic stem cells. So I'm showing you here on the left the editing activity in both a plerixafor mobilized normal donor, as well as 3 sickle cell patients, 3 donors that are patients with sickle cell disease. I'm showing you the editing in the input cells, the bulk CD34 cells, in which we can achieve pretty high editing levels, around 80% or so. And what was particularly encouraging to us from this data was that 17 weeks after transplant into these NBSGW mice, we could see pretty persistent retention of the edits on the level of between 60 to 80%. In addition, if we're looking here on the right here, I'm showing you the amount of fetal hemoglobin that can be induced. So this is, again, after transplant, harvesting the erythroid cells from the bone marrow of these mice. You can see that particularly in these sickle cell patients, we're able to achieve levels of around 30% HbF induction, which was we were quite happy with. And so we think that this is a pretty promising therapeutic approach in which we're trying to perform a number of IND-enabling experiments characterizing a scaled-up manufacturing process where we're trying to characterize the pharmacology and toxicology of these to bring these to a clinical trial in the next year to year and a half. But one question that we and others have here is, like, how do we understand the safety of these types of really promising genome editing approaches? So typically, hundreds of millions of cells are modified by therapeutic genome editing. In the case of this hematopoietic stem cell therapy that we're working on, we require at least 2 million cells per kilogram of patient body weight to get efficient engraftment. And so you're looking at editing of 150 to 200 million cells potentially, depending on the patient, if we're starting with young adults. These edited cells might persist for years to a lifetime. And even low frequency off-target mutations might be relevant if they induce some sort of cellular growth advantage. So I think here it's really important to learn the lessons from gene therapy. One guiding issue is that in early gene therapy trials for severe combined immunodeficiency using gamma retroviral vectors, this trial resulted in T-cell leukemia in about 5 of 20 young patients. And so when it was looked at in more detail, it was found that it was really vector integrations near the ELMO2 proto-oncogene that were really causing and driving this T-cell leukemia. And so this ended up being a really tragic setback for the gene therapy field that really set back things by about 10 years when people were really trying to figure out the safety and improving the safety of these types of viral vectors. And so this is the type of thing that we are also really thinking about in terms of advancing genome editing therapies. And so this is a question that we've asked over the last several years. What's changed with genome editing? And so this is a slide, I think, that also summarizes potentially like 5 or 6 years worth of work where we're thinking about defining the genome-wide activity of genome editors. I thought it was nice and was pleased to see Trevor put up a cartoon that I often show about what's missing about the streetlight effect. And so that's kind of illustrated over here on the left, where it reflects the natural human tendency to look where the light is better, for example. So if you're looking for your keys in a dark parking lot, you might tend to look under the light. And so this is very similar. If you imagine that the genome is, if the parking lot over here is the genome, it's very similar to the idea of using targeted sequencing, for example, and computational prediction to try to identify unintended effects of genome editors. You might be able to find a few, where the light is better, where you're performing these targeted sequencing experiments, but you might miss a number of other sites of bona fide activity simply because you're just not looking there. So that's a way to illustrate the limitations of targeted sequencing. It's also possible to use methods like whole genome sequencing, and I think that's something that people initially think about, but I think the analogy here might be shining a very dim light, for example, on the parking lot. So whole genome sequences are very broad, but they're relatively shallow. And so they're not really sensitive enough to be able to identify genome-wide activity where we're interested in effects that are as low as 1 in 1,000 or much below that. So several years ago when I was a postdoctoral fellow in Keith Zhang's lab, I developed a method called GuideSeq, which is really based on the principle of efficient tag integration into the sites of double-stranded breaks in living cells. And so it's been really rewarding to see this method used quite broadly in the development of genome editing therapeutics, where I think it's one of the most widely used methods. However, I think some of the limitations of GuideSeq are first that sensitivity is probably limited to around 0.1%. And so if you really want to have a much more highly sensitive method, this method becomes a bit difficult to scale because it is a fairly quantitative method. And so if you were to try to scale it, you would need to scale both linearly in terms of the number of cells that are being used as well as the depth of sequencing. Another limitation is that it's not necessarily, it can be difficult to apply to certain primary cell types. For example, harder to apply to human hematopoietic stem cells, for example. So that's kind of motivated the development of in vitro methods that can be highly sensitive that I'll tell you more about here. So when I started my lab at St. Jude, we really wanted to develop a highly sensitive, unbiased in vitro method. This method, I guess, is based on some earlier methods that I had developed called CircleSeq. And so our wish list for these high throughput methods were that we really wanted to minimize genomic DNA input requirements. We wanted to simplify the workflow. We wanted to have really low hands-on time and cost. We wanted to achieve equal or better sensitivity than earlier methods such as CircleSeq that I mentioned, and increase the scalability and throughput. And so the method we came up with here is called circularization for high throughput analysis of nucleus genome-wide effects, or ChangeSeq. And so we were really able to use TN5 tagmentation to really streamline the workflow of an earlier method that I mentioned called CircleSeq. The principles are the same in that we're trying to really selectively sequence Cas9 cleaved genomic DNA. And so the idea is that we've developed a really streamlined process to take genomic DNA, to tagment the DNA, to circularize the DNA, and degrade away residual linear DNA. We take CRISPR-Cas genome editors, we treat this population of highly purified genomic DNA circles, and only circles that are cut will have ends that are available for adapter ligation and sequencing. So when we first embarked on this project, I told my really talented postdoctoral fellow, Cesar Lazarate, that led the development of this work, that this would be a relatively straightforward process. But it turned out that it actually did require a pretty extensive optimization. So over the course of, I think, 20 or so sequencing runs, these 42 samples, we were really able to finally achieve enrichment of the on-target read counts comparable to our earlier CircleSeq method. Over here on the bottom, I'm showing you an image that was provided by our collaborators at the National Institutes of Standards and Technologies. They really surprised us with this atomic force microscopy image, which is really direct visualization of ChangeSeq circularized DNA molecules. And so what you can kind of see from this field is that by this direct method, we don't really see evidence of linear DNA. So suggesting that you can really have highly purified circular libraries that are required for this method. So comparing ChangeSeq to earlier methods such as CircleSeq, we can see that ChangeSeq is really comparable to the earlier methods in terms of the sites that can be detected and the correlations. You can see they're really quite good. The method is also highly reproducible in the sense that, like, we're looking at here, for example, 12 different target sites where we're performing technical replicates for ChangeSeq. You can see that most of the R-squareds are above 0.95. So we wanted to perform high throughput profiling of genome-wide activity. We're doing this now in human primary T cells because this is a really accessible cell type for us. It's commonly used for therapeutic applications, including cancer immunotherapy. And importantly for us, it's a cell type where we can perform both guide-seq and change-seq. So here I'm showing you data from some of these change-seq experiments. These are Manhattan plots where you can see we've got three gene targets in pairs. On the top, I'm showing you a highly specific target. This is a Manhattan plot where you see genome-wide activity organized by chromosomal location. You can, and on the y-axis, we have change-seq read counts. The on-target site is marked by a red arrow, and you can see that in this case, we have 3 cases where you have really highly specific guide RNA targets, while below you can see much more promiscuous activity. And so using change-seq, we were able to profile 110 of these therapeutic target sites, finding over 200,000 different unintended off-target sites. And I think it's important to note here that there are some that are quite specific, where you have either single on-target targets or very little off-target activity, as well as those that are quite non-specific. So here I'm showing you a plot of the change-seq specificity ratio. So we basically define this as the ratio of the intended on-target activity to the sum of all genome-wide activity. So higher change-seq ratios mean that this particular guide RNA target is more specific. We also edited these 110 sites in human primary T cells in triplicates. And so here we're seeing the average of the actual editing activity. And so from this plot of editing activity against specificity, you can see that you can really identify these targets that are both highly specific and highly active. And so that's encouraging for therapeutic application. So we also wanted to ask the question of what we could understand from these large-scale change-seq data sets. We wanted to understand whether there are any specific sequence factors that are associated with unintended genome-wide activity. And so we asked whether A, C, G, or T base frequency in the target site was associated with genome-wide activity, as well as the number of sites that are detected, whether nucleotide diversity or RNA structure were also associated with the number of sites of genome-wide activity detected by change-seq. The 2 effects we pulled out that were particularly striking are for G-based frequency, where we found that with increasing number of Gs in the CRISPR-Cas target sites, that we detected a larger number of change-seq sites. Additionally, if you had more nucleotide diversity in the target sites, you would have kind of the reverse relationship where you have less genome-wide off-target activity detected. I would mention that those observations are pretty consistent with the idea perhaps of a wobble-based pairing between the guide RNA. And so this is something that we've previously observed, but with less confidence in the guide-seq data set, which others have also seen. One question we also wanted to ask from these large-scale change-seq data sets was whether we could use machine learning to really begin to predict off-target activity. So there's been a lot of work in terms of off-target prediction in the past, including by one of our speakers, John Dench. I think there's been really fantastic work in this area. I think one of the limitations is that a lot of it is based on limited genome-wide activity data sets, such as the ones that I'd worked on in guide-seq in the past. So based on just 10 or 15 sites. And so the idea is if you can have higher quality, larger scale input data, can you train better machine learning models? I'm not going to go into the technical details of this machine learning model here, except to say that it's kind of based on on the ensemble of different weaker predictors called decision trees. I think one of the important aspects of this is that not only is it's not necessarily just a black box predictor, but it can also give you some insight into the features that are important for the prediction. So on the top, we can see that this machine learning model seems to perform pretty well in terms of both ROC curves and precision recall. One interesting note that came out of it was that we found that in terms of feature importance, the positions 2, 10 through 12 seem to be particularly important. And there were some structural support for some of this this possibility where there are some contexts to the DNA at those positions. There's also, again, we see this G to A, and we think that this is these G to A mismatches being really important features. And we think that that's because of the tolerance for RNA G to DNA T base pairings. We wanted to validate the sensitivity of change-seq. We had done this initially by guide-seq, and we were also asked by our viewers pretty reasonably to do this by targeted sequencing. So we used a method called RAMP-seq to perform multiplex targeted sequencing of over 600 different on and off target sites detected by change-seq. We note that we have a particularly high validation rates for those detected by both change-seq and guide-seq, but that there were about 20% of sites, here we've divided them into different classes, that could be detected exclusively from the change-seq set. So they're discovered only by change-seq and not by guide-seq. We're using a really sensitive tag integration sequencing method to detect these sites. And I think it's really important, I think, in studies of trying to understand genome-wide activity to take into account the sensitivity of your validation methods. If you're not using highly sensitive methods, you obviously won't detect some of these low frequency sites. And so I think that it's important to take that into consideration when declaring something as a false positive or false negative. Overall, we're able to validate at least 20% of the sites that we're able to detect by change-seq. We find that it's more sensitive than guide-seq for detecting bona fide genome-wide activity. One question we're interested in also is what is the relationship between the cellular and in vitro measurements? So change-seq, of course, is an in vitro biochemical method that we're performing on naked genomic DNA. And so to try to understand the impact of epigenetic features on this, we took human primary T cells, we stimulated them, and we performed a number of these parallel measurements. We had guide-seq, which I described, which I mentioned to you before. So we're performing guide-seq on a large number of these different targets. We're also in parallel performing ATAC-seq to understand chromatin accessibility, ChIP-seq to profile histone modifications. So these particular different histone marks. And the idea here is that there is a large set of sites that are potentially detected in vitro. This is highly sensitive, but some of them might not really be cleaved in cells because of other chromatin factors. There are the sites that we can detect by guide-seq in cells. This is probably like the top 99% or so of sites. It's not as sensitive as in vitro methods, so you're missing some bona fide sites, but perhaps if we can understand how chromatin states, chromatin accessibility impacts which sites are being cleaved, we might be able to better predict in the future the bona fide sites. And in the long term, we'd like to know which ones have a real functional impact. Okay, so here we're looking at some plots I'm showing you of some histone modifications here, H3K4 trimethylation, ATAC-seq, and H3K27 acetylation. These were the ones that were most strongly associated with genome-wide activity. And so I'm showing you the enrichment for either guide-seq sites, which we see on the top, versus change-seq and Cas finder. And these are in silico predicted sites or random sites. And so for these particular marks or the ATAC-seq track, you can see that there's really a strong enrichment in guide-seq in these marks at guide-seq sites, which really represents the sites that can be detected in cells. And so these marks are associated with active chromatin. We wanted to look at this even further in kind of a way inspired by the ENCODE project. And so we took our histone modification ChIP-seq data and we used to really partition the genome into a number of different chromatin states. And so these partitions are based on the combination of different marks of these histone marks and or ATAC-seq. And so you can see we partition the genome into 25 different chromatin states, and then we're trying to determine whether or not there's enrichment or depletion of change-seq or guide-seq sites. And so what you can see is that if you're looking as expected with change-seq, since the in vitro method, you're not really seeing much enrichment for any particular chromatin state. But if you look at the set of sites detected by guide-seq, these are cellular off targets, you can see that there's a pretty good enrichment for active promoters, sites with chromatin states of strong transcription and active enhancers. And so essentially cellular off targets are probably about 2 to 4 times more likely to be found in these types of chromatin regions. It doesn't exclude the possibility that there are off-target sites that occur also in heterochromatic and other regions, but they're enriched in these particular chromatin states. One last question that we wanted to ask from the change-seq data sets is, does human genetic variation really affect the genome-wide activity of these CRISPR-Cas genome editors? And so here we're really leveraging really well-characterized genomes characterized by the NIST Genome in a Bottle project. So we're looking at 7 genome in a bottle genomes. We're looking at 6 targets, 2 replicates, with 84 change-seq runs. And so this is really enabled by the scalability of change-seq. In this here, I'm showing you a volcano plot where we're looking at the effects of genetic variation on genome-wide activity. And the y-axis is statistical significance, on the x-axis the effect size. And so we find about 15% of sites had where we observed a significant effect of genetic variation on genome-wide activity. To look at this in a little bit more detail, I've kind of shown you 4 examples in much more detail. So we can see these are examples where there's a clear quantitative relationship between the genotype and change-seq read counts that you can see on the x-axis. So depending on which genetic variant you have, whether it's homozygous for one allele, heterozygous, or homozygous for the other, you can see some increase, for example, in change-seq read counts. We also wanted to look at this on the direct allele level. And so we're looking at also whole genome sequencing and change-seq data. So we pulled out certain heterozygous sequences. So these are 4 different off-targets. And so we have an individual we're looking at the activity in a heterozygous individual. In the whole genome sequencing data, as you'd expect, you see a 50-50 allelic ratio. In the change-seq data, we can see pretty clear evidence of allelic bias, showing you that there's clear effect of the genetic variation on the activity. And some of these make a lot of sense, where instead of an NGC, for example, in the PAM sequence here, you have an NGG. And so you have activity only when you have that particular variant. And so that suggests to us that there's the potential for using change-seq potentially to screen for really highly specific targets. You can validate them for both on-target and off-target activity. And that in the future, it might be important to consider this kind of genetic variation when you're going into clinical trials in patients. I think one important question is whether these sites where there's effects of genetic variation in vitro, in a biochemical sense, are also really reproduced in cells. And so that's something we're also thinking about. And so to take it really full circle, here, again, I'm bringing you back to our plan to advance the CRISPR-Cas genome editing as autologous human hematopoietic stem cell therapy. At our gamma globin promoter target, we're looking at genome-wide activity. And for this particular target, so far, the top off-target sites, we see that it's pretty specific. And we're continuing to characterize this. And so as a summary, I think that there are clearly very exciting prospects for therapeutic editing, both for sickle cell disease and other disorders, that it might be important to consider effects of human genetic variation. I think this is a pretty important open question. I've shown you more advanced genome-wide methods that are really simple to perform, highly scalable to really help see the big picture. So methods like change-seq. I think an important future direction for us is to also understand the biological consequence of some of the unintended mutations, this unintended genome-wide activity. And so that's something we're thinking a lot about. Overall, because I think sometimes when I give talks on genome-wide activity, the inclination is to think that this is a terrible thing, that we can see a lot of off-target effects. I think that instead I'd like to bring a glass half full approach and say that this is really a reason for optimism and confidence in therapeutic genome editing. And with that, I'd like to thank members of my lab, our funding sources, the Doris Duke Foundation, the NIH Somatic Cell Genome Editing, to mention very briefly that we are hiring, and I'm glad to take any questions.
Thanks, Jinder. I don't see any question for you. Can I ask a quick question? So for therapeutic purposes, what do you think is the tolerable level of off-targeting?
I think that's a good question. I think that it's not easy to answer, and I don't think there is necessarily like a specific number. I think that sometimes people point to a threshold such as, okay, this is the level of somatic mutation rates in cells, for example. And I don't really think that it makes sense to declare an absolute number. I think that, for example, if you had a mutation in p53, that it doesn't matter necessarily the frequency. I think you'd still be concerned with it even if it was like 1 in a million cells. So I think it really depends on the functional impact of the particular mutation. And that's something that I think is not easy to do right now. I think that for any arbitrary unintended mutation or any arbitrary sequence of the human genome, it's not easy to know what its function is right now, right? And so I think that right now we tend to just try to minimize off-target activity, but in the future, if we want more confidence, I think we'd also like to be able to interpret it.
Cool. All right, we're a little behind, so unless someone else, speakers has a question.
I have a quick question if you don't mind, just a technical question, Shaga, great talk. I'm curious why when you were using TM5 fragmentation at the tail, why did you want to have the user treatment extra step instead of having a sticky end directly? Just curious.
Oh yes, yeah, that's a good question. I think in terms of that optimization, that was one of the things we tried was to directly have a sticky end. I think there's some technical issues with the processing of those. So you do have to perform gap repair with TN5 fragmentation because it does leave a 9 base pair gap in the other strand. And so in the process of doing that, you have to have some method to maintain the end, and the polymerases that are doing that will often either blunt or chew off the end when you're performing the gap repair. And so that's why we ended up having to use user enzyme to create.
Okay, that makes sense. Thank you very much.
Yeah, sure.
So you have a question?
Yeah, I have a quick question. So have you compared like those change seed, they are in vitro validation. Have you compared like in vivo, like maybe do the editing in vivo and isolate those cells, and then compare the off-target profiling, like, you know, to show like, is there any correlation between the in vitro and in vivo?
Yes, I think that was actually the data that I was showing you in terms of the targeted sequencing validation. I think I might have gone over it kind of fast, but so that was basically in edited human primary T cells, edited with Cas9 ribonuclear protein complexes and no particular tag. And so we're basically in the presence of a guide-seq tag, and we're basically doing targeted sequencing in edited human T cells to confirm that about, I think between 20 to 40% of the sites that we detect by change-seq can be validated in this way.
So there are a couple of questions in the Q&A box. One is, have you thought about the possibility of reducing NHEJ events to more HDR to reduce off-targeting?
Yes, so I mean, I think that this is definitely a question that we and others in the field have been thinking a lot about. I think one of the challenges is of doing this in kind of a target-specific way. You know, there are broad inhibitors, for example, of DNA ligase 4. For example, to do this, some of them with varying levels of efficacy. And there are also fusions of Cas9 to other factors that might recruit HR factors. So that's something we're definitely thinking about. And of course, base editors, if your target site is compatible, is another good option for being able to make some of these conversions.
Okay. Another question is, could you comment on false positive rate of change-seq?
Yes. So I think that as an in vitro, the false positive rate, I think is something that's a little challenging to understand. And I think this is the point I was making about highly sensitive sequencing. So for example, I think, there's been therapeutics companies that have been comparing, for example, guide-seq, circle-seq, which is, predecessor to change-seq. And I think they basically came to the conclusion that guide-seq had a much lower false positive rate, and I guess as someone that's worked in both technologies, I guess, it's nice to hear something like that. But I think it's important to critically scrutinize these kinds of conclusions, because I think they're basically, in that study, they're basically declaring that targeted sequencing is the ground truth. That if you don't detect it by NGS, which has a limit of detection of around 0.1%, then they declared everything else is false positive. And I think it really depends on the sensitivity of that. I think we found that at least 20 to 40% of the sites we detect by change-seq can be confirmed by really sensitive targeted sequencing, where we can get down below 0.1%. But I think the true false positive rate is really not yet known. It could be better. It could be less than that.
Thanks again. We will go on to our next talk. So next speaker is one of our own, Tim Peterson. He's an assistant professor here at WashU for internal medicine, bone and mineral diseases and genetics. And I'll just say that Tim has a really unique and intriguing angle to study aging. I'll leave it like that and have him tell us the story.
Thanks so much. Can you hear me all correct? Okay, great. Thanks. So yeah, so hopefully you can probably tell from the title of this talk, it's be a little bit different. I'm definitely much more of a technology user than technology creator. We tend to focus on the biology, but I'll try and tie it together with, this kind of very technology development seminar. And so thanks for having me. So the title is Sphingolipids Connect to Pathogens to Aging, and I'll go through how we got to that title. So just to kind of outline, I'll break it down into essentially kind of three parts. So how we get to sphingolipids is with a collection of drugs that we call CADs, and I'll run through those in a second to explain to you why we got interested in these drugs. Of course, pathogens, and then lastly, aging, and I'll tie those three together. And then lastly, I'll say, how does this relate to this conference? Okay, so CAD drugs we've just given them this name, CAD drugs. I don't, it's not commonly made into an acronym, but essentially it stands for cationic amphiphile drugs. So they have these chemical properties of being cationic amphiphiles. They're also known as lysoosmotrophs because they have a tendency to home to the lysosome. So the reason we got interested in these drugs is because they're some of the most widely used drugs really on Earth. 20 to 25% of all FDA-approved drugs have these cationic amplifier character. So just on the left and the right, I'm showing a collection of very commonly used drugs, both non-CADs and CADs. So non-CADs on the left and CADs on the right. So you may be familiar with atorvastatin, which is one of the statins used to lower cholesterol. Metformin is one of the kind of most well-known diabetes drugs. Alendronate is one of the most popular osteoporosis drugs, and one where we spend a lot of time studying. And then there's a variety of CAD drugs that you may have heard of. Tamoxifen, which is well known for its use in breast cancer. Chloroquine got kind of made infamous due to COVID-19. And there's a variety of antidepressant drugs, Prozac, which is fluoxetine, and sertraline, which is Zoloft, and others that all have this cationic amplifier properties. And so just to kind of be more precise, essentially what we mean in the medicinal chemistry world, we mean that that CAD drugs have a basically high C log P, which is basically oil to water partitioning. So greater than 2 is tends to get you in this kind of CAD territory. And then they also are weakly basic. So they all have a pK of, it's actually said to be maybe greater than 6.5, kind of puts them in this category. So you can see kind of how all these break down above that threshold. And whereas other non-CADs do not have these characteristics. So our basic question was, well, can we do screens with these drugs and try and understand them a little better? So I just said this out loud, but just to kind of again reiterate, we really just focused on these two parameters, the C log P and the pKa, oil to water, how basic it is, and we said, okay, a lot of these kind of famous drugs have these properties. What would they look like in a screen? It was pretty much pretty simple approach. So we did our kind of screen strategy of choice is used the CRISPR-I. I know the traditional CRISPR works quite well, but we were quite happy with some of our initial results and worked with Jonathan Weissman and Luke Gilbert on this method. We definitely became kind of disciples of this approach. So really and you probably are all familiar with this from what Trevor talked about earlier, but just to reiterate, mutagenized whole genome screen, and mutagenized cells, we tend to stick in one cell type. So K562 cells, kind of our Saccharomyces cerevisiae that we tend to work on. And then it's just a fitness-based screen. So treat with drug, watch the cells grow, deep sequencing. So quite basic, and just looking essentially for gene-drug interactions. Get a lot of questions about why you would ever do this. If you're talking about antidepressant drugs and cancer cells. But I think hopefully I'll convince you that we are able to get meaningful results out of these experiments. So we've done tons of screens with a huge variety of drugs. I think what was really kind of exciting was when we did a meta-analysis of several of these screens. So if you took the 6 CAD drugs that we had screened at that time, and just kind of said, oh, what's the z-scores basically for these drugs for the top hits. Essentially, all of them broke down as genes related to sphingolipid metabolism. So so of course the kind of initiating enzyme in sphingolipid biosynthesis, which is serine palmitoyltransferase, was our #1 hit. And there's a variety of other enzymes in this process which are important, which were really at the top of the list for all these drugs. And then other genes and other protein complexes which are involved in trafficking, but when disrupted, have huge kind of sphingolipid phenotypes also scored, such as the GARP and COG complex. So it really was a very specific signature with all 6 of these diverse CAD drugs, so that they all have widely different therapeutic uses. Okay, and then what was also great to see was that these results were incredibly specific. So we've lined up many of our non-CAD based screens as well as screens from other people, and the pattern is just kind of night and day. It's just, when you look at what scores with these with a CAD drug versus a non-CAD drug, you can see it's just, like I said, it's pretty much black and white here. And so this is just kind of a heat map of the z-score results. Okay, so just a quick summary of that, which was pretty exciting to see that this was just a simple idea, unbiased approach. Was that we identified the sphingolipid-related genes. The genes that we identified in screens using the CAD drugs all kind of interact with sphingolipid-related genes, and so that suggests that potentially when you use screens with drugs that that share chemical properties essentially with the kind of metabolic machinery that they interact with, that you'll see those in the screen. So it kind of suggests kind of, again, a gene chemistry genetic interaction there that pops out pretty easily from these screens. And then I think maybe a broader point gets into this question. Well, we thought we knew what all these drugs did, but now our screens essentially are telling us, the antidepressants are involved in serotonin. Well, if you and the other drugs have other mechanisms, but these drugs really, these screens really suggested that potentially the mechanism of actions of these drugs may need to be reinvestigated. So Jason Seltzer at Cold Spring Harbor has done some work on this with cancer drugs. So our work really kind of parallels that line of investigation that he's on, and I have a link to his paper here. Our work's still on bioRxiv, but yeah. So I think these screens really can open up I think some new directions and really force us to reimagine how small molecules are maybe conceived of. Okay, so pathogens. So there's been a lot of CRISPR screens with pathogens, of course, most recently with SARS-CoV-2. So we, and in general, when people do screens with pathogens, what you end up seeing is, there's some hit, often the receptor, and there's kind of a story made about how that particular protein, for example, the ACE2 receptor in this case of SARS-CoV-2 is how that particular protein is interesting. But we asked a different question. I was very fortunate to kind of in my training to be around Tyne Brumelkamp and Jan Corrett, who really, I think, pioneered a lot of the early efforts to do genome-wide screens with pathogens before the CRISPR technology took over. So a lot of these publications that I'm citing here on the left here are from them, but there's of course many other folks as well. So I just want to highlight their kind of seminal role. So we asked a different question. We said, okay, what may be the common features, the common host factors that could be important across a wide variety of pathogens. So we took, all the basically available to us virus screens, screens involving bacteria or toxins from bacteria, plant toxins and other parasites. And we said, what are the host factors, the human genes that are mammalian genes that are most important across a variety of pathogens. So when we did this experiment, or not experiment, we just simply kind of organized the data. it's essentially the exact same list of genes. It's the sphingolipid related genes, and particularly the GARP and COG complexes that so strongly regulate sphingolipid metabolism. So basically, what we're just showing here is the the p-values, kind of a way of kind of normalizing the p-values across these screens on the x-axis, and then the kind of an aggregate measure of the z-score, if you will, the kind of the effect size, essentially the number of screens where the pathogens, where these genes, number of screens where these genes scored on the y-axis. So again, the list is pretty much the same cast of characters. And then if you line it up with the CAD screens versus the non-CAD screens, again, it's pretty much a kind of a black and white pattern here, where the CAD drugs and pathogen screens really have a very specific gene signature. So this suggests to us, well, it seems like sphingolipid related genes are particularly important to a wide variety of pathogens. And then so the question for COVID-2, I know now we're into the vaccine era, but kind of before that, we were saying, well, shouldn't they have been tested? Our work came out around March or April, and was on bioRxiv, so it didn't really, wasn't really kind of immediately helpful. But there was chloroquine that failed. So chloroquine is actually a CAD drug. And I'd actually argue, if you look in I think most people that have some knowledge about this, it does work quite well in vitro. The question is, I think due to its effects in the heart and other tissues, I think it just doesn't have a very good therapeutic index in vivo. But on the other hand, other drugs are showing promise, kind of most notably a trial out of WashU. There's a drug called fluvoxamine, which is an antidepressant drug, which Eric Lentz and colleagues have shown in a recent JAMA article, which I'm linking to, is showing promise in terms of people testing positive for COVID. So there is evidence that that a CAD drug could be actually quite valuable, and they're going on to expand this study to because of the ease of use of antidepressants. So. All right, and then so then where do we go from there? So now we have this connection between sphingolipids and pathogens. What next? So we wanted to be to think beyond just CRISPR. I know this is pretty much primarily a kind of a gene editing conversation, but of course there's other kind of genomic methodologies. So here's again just quite basic type of analysis. Does not take it's not fancy technology today. So we just simply compared transcriptional profiling when treating our Saccharomyces cerevisiae K562 cells with CAD drugs or non-CAD drugs. And the pattern here was also, if you did this kind of meta-analysis of the CADs versus the non-CADs, was also, just produced this very specific signature that was unique to the CAD drugs. In particular, really all the top hits were all genes that had been linked to cell senescence. And so you may or may not have heard a lot of these, but of course, like P21, for example, is the kind of famous target of PBD3. SERPIN1 has quite a lot of interest. It's known as PIA1. And a lot of folks have studied that gene. IL-6 may be familiar to you all, one of the tumor necrosis factors. So there was a huge, long list of some of the top kind of senescence regulated genes in using these CAD drugs. And if you look in the literature on what causes senescence, certainly sphingolipids and ceramide, which is one of the kind of most well-known sphingolipids, is kind of right at the top of the list of kind of causal agents in cell senescence. So that made us pretty happy. And then and other things of kind of quality control genes which are involved in sphingolipid metabolism were down-regulated. So it really suggested that these drugs were having an on-target effect on sphingolipid metabolism. And again, using a different modality than CRISPR screening. Now we're using just RNA-seq. And then, so the question is, okay, so here's kind of our gene signature, transcriptional gene signature with CAD drugs. How about the pathogen gene signature? So that's actually basically identical. It's really the same cast of characters. Yet again, the elongases are downregulated and the P21, SERPIN, IL-6, all these factors really are right at the top of the list amongst the genes that are kind of universally regulated, if you will, by pathogens. So essentially didn't matter the context. We took, mice, rats, basically we grabbed experiments from all over the place. We profiled 13 different pathogens across 30 different data sets. And so the pattern is really quite robust. This again suggests a strong overlap in the biology here between the CAD drugs, sphingolipids, and pathogens. And then moreover with cell senescence, which connects us to aging. So kind of in summary from this part is that CADs and pathogens regulate senescence-associated genes. And then shouldn't we be testing CAD drugs for aging-related diseases? So we've actually already done these experiments. The data is still a little bit rough around the edges, but essentially one of our top hits when we asked the question, which drugs would regulate these transcriptional signatures the best? Essentially, it's a class of drugs called senolytics, and these are drugs. So most famously, dasatinib and navitoclax are both CAD drugs. So these are drugs. There's a whole related field called senolytics where people are trying to see which target just the senescent cells themselves as a strategy for preventing aging. And sure enough, those are really right at the top of our list when we do experiments looking at pathogens and profiling pathogens and which drugs may regulate them. So that's, to me, suggests that it kind of closes the loop here in saying that at least one mechanism by which we could potentially treat aging would be to just continue the strategy that people are following with the senolytics, but one of the, I guess, the twist here is that this also implicates pathogens in the aging process, which is maybe a little less appreciated. Okay, so my general points about what this all has to do with this conference is maybe I'm kind of preaching the choir here, so I'll totally admit that. But I think this is important to kind of dwell on just for a second. I come from the RNAi era, and of course, I still love RNAi, and Alnylam and other companies are really doing great things in that world. But I think what we're talking about with gene editing is, at least in our case, we think we identified some pretty key biology, which was made possible by kind of very robust methodology that a lot of you are creating. And so I think we're really at a inflection. We're really at a point where the robustness of the technology is really enabling us to see patterns that were not previously kind of unattainable to see. And by simple kind of analysis where you're kind of just aggregating data together. And so you really, it's the whole garbage in, garbage out. If the data is not good to start with, you're going to get kind of garbage out. And so I think, thanks to all of you, in particular, Luke and Gilbert and others, that we're not dealing with garbage in, garbage out now. What we're putting in is highly, high quality data. And that's what gene editing technology allows us to do. So both kinds of in terms of discovering biology, but also in terms of modifying. In our case, we'd argue, well, we're not really modifying, we're talking about drugs here, but essentially idea is how you would modify it. And what we're suggesting here is that these CAD drugs could be important, but you can imagine manipulating the genes directly with the gene editing approach. So in terms of the wet lab experiments, we think again, preaching to the choir here about how how we can do experiments that are unbiased, so you really get the key things, and that they're when you get your results, they're highly accurate, and that you have high confidence that what you're pursuing is important. And then just like a lot of us, so we spend a lot of time doing these screens. They're actually pretty simple, but we want to be able to do, there's roughly 2,000 FDA approved drugs. We'd like to do all of them, but we're not a big institution. We're just currently a small lab, and so the question is, just some examples of people that are kind of moving closer to the clinic are Verve Therapeutics. They've got some great data with CRISPR approaches. And then there's a really nice study where they corrected using CRISPR-A in mice that came out of UCSF, I think a year or two ago, where the CRISPR-A effect was about 6 months. And so these are just two of many examples that I particularly liked that you could investigate were kind of very very potent effects we're seeing. Okay, and so in terms of what we're trying to do, so we're trying to move away from doing experiments. I think the power will be, essentially what can you predict without, again, if what you have is accurate, you even want to just make it, you basically want to be able to do more experiments. And so of course, computers are the way to do that. So our kind of inspiration for that is that we started here with, we have in machine learning world, you talk about inputs. And so here now we have two inputs, C log P and pKa, and they can predict, the screen results for, we think for any drugs. We've now tried this with a few more drugs, more than the 6, and sure enough, the patterns just exactly the same. And so we think this really would extend to all, the several hundred FDA approved drugs. And so potentially all those mechanisms, whatever they are, may not be accurate, and what we're proposing may be accurate. And then another inspiration for this is the depth map. So this really enabled us to understand gene-gene interactions really without doing a directed experiment on any particular gene. And so we would like to, our focus is on kind of drug-gene interactions. And so can we do this again without doing any wet lab experiments? And so I just like to thank a lot of people, particular Luke Gilbert, and we've got a company that's kind of starting to do this, do these experiments, both the wet lab and then transitioning to the dry lab. And so that's pretty fun, and just thanks for your time.
Thanks, Tim. Anyone has questions for Tim? Speakers, you have to turn on your the microphone to ask a question. All right.
Sure. Yeah, I actually have one. I'm sorry. So I think this is absolutely fascinating, and in particular the lipid biology aspect of the whole thing. On a related, or hopefully related note, we see in K562 and a couple of other AML cell lines sort of a rewiring of lipid metabolism that's not really consistent with some of the rest of the AML cell lines. So we find 4 that have that are particularly sensitive to saturated fatty acid species, and the rest of them are not. And we don't really understand why. So that's a big sort of open question that we're not really terribly well equipped to understand. But I wonder how consistent these results are, if they're in a single cell line, or how diverse might they be in different backgrounds?
Yeah, so we actually haven't. That's a very good question. We haven't tested that. Some of the companies we're working with are, of course, very interested in that. Like I said, I'm touting this as kind of the K562 is the Saccharomyces cerevisiae. It is for us. But so yeah, the extent to which you really can rely on one cell line and kind of make extrapolations to other contexts. We're testing is the short answer with breast cancer cell lines right now. So I can email you about that.
I'll touch base with you about our story as well.
Yeah, so that's a big question. I mean, certainly. I mean, we've tested in small scale. That we of course did follow up, knock down the SPT gene and a few others in breast cancer cell lines, and of course the phenotype is still there. I mean, what we can't say yet, but should know soon is is SPT number 1 hit across in other cell lines like it is with the K562 cell. So I'd imagine it'll be close to the top, but how top. So yeah.
Here's a question from Kyung Hee on the topic of aging. You suggested that the response to CAD is the senescence, but the genes that you're looking at are the inflammatory response genes. Is it possible that the sphingolipid pathway genes are actually involved in the inflammatory response, and you are actually examining sphingolipids in inflammatory response?
Sure. So I mean, this kind of gets into like terminology and human annotation. So yeah, we originally kind of were, I mean, as you can see, IL interleukins and a lot of them have kind of inflammation, and that of course makes sense too with the with the pathogens. but if you look, it's there's a lot of cell cycle regulators in there too. So it's really, the signature is much more of a senescent signature. But I think more or less this question becomes, which gene annotation software, or which kind of, what's your, worldview on, what categories these genes fall into. In our view, the dominant signature is one of senescence, and so these cytokines are certainly part of senescence mechanisms, and the SASP, as they call it, senescence-associated secretory phenotype. So it's all kind of the same stuff, if you will.
Thank you, Tim.
Yep, thank you.
Next speaker is John Dench. He's the Associate Director of the Genetic Perturbation Platform at Broad, and he's also on the advisory board for Epigenome that we almost use every day. For those of you who work with us on CRISPR reagents or cell lines, you've seen in the reports on the guide RNA design slide, we have a column called Dench scores. So that's for prediction of guide RNA activity. And I just want to say that Dench is this Dench. And we included that in there just in case something doesn't work. We have a big name to blame. Fortunately, that doesn't happen very often. So John's title is Massively Parallel Variant to Function Mapping with CRISPR-based editor screens. John?
Great. Thanks so much. I assume you can see my slides?
Yes.
Okay, great. Just by the way, I've never personally called it the Dench score. I don't know where that came from. I call it rule set 2. I don't like naming things. Okay, here are my disclosures. It's so nice to see so many wonderful people from the community. I don't know, I woke up with a real sense of optimism today. Who knows why that could be. So I'm gonna talk about our work with base editor screens. Now, Ibrahim gave a wonderful introduction to this, so I will certainly not belabor in any way. The basic idea here is that people have appended various things to Cas9 to allow it to, instead of cutting DNA, or instead of binding to DNA and then modulating transcription or epigenetics, here we're doing chemistry on the base itself. Now this review from Holly a couple of years ago already presented that there was a huge diversity of base editors out there, and that has only continued to grow. And of course, we're going to hear from Andrew next about prime editors for the next step in the evolution of this technology. But what I'm going to be presenting today is our work with what was called at the time base editor 3, that has an APOBEC domain at the N terminus, a UGI domain at the C terminus, and allows the conversion of C to T. So the question we were asking is, can we turn this into a scalable, screenable technology? So sorry about that. So the first thing we did was we made a library that tiled across a whole bunch of genes. And we did some straightforward viability screens that many have talked about already. And for this first test, we did it in hap1 cells, which are a haploid cell line. So the first thing we look at is when we have guide RNAs that are targeting the heart-annotated non-essential genes, how many of them deplete? And we draw a threshold at about 5% and say anything to the left of that is depletion. So we're using our negative controls to set that threshold. Then we can look at the guide RNAs that now that they're intended to target an essential gene, but either the prediction is that there's no edit because there's no C in that window, or it's a silent edit because of the wobble, is editing the wobble position and not changing the codon of the protein. And we see a little bit of a shift to the left. Relative to that 5%. So there's some sort of unexpected editing occurring, and that could be due to a couple different mechanisms. First, out of window editing. So, all these base editors have a specific window where we think the editing is occurring, but it's clearly not, from the plots that Ibrahim showed, it's not like nothing, 100%, 100%, and then nothing. It's, a peak of activity in some shoulders. So it could be editing in the shoulders. There could be some indels occurring. We intend these things to not introduce indels, but they could. And of course, especially for the silent mutations, it's worth remembering that not everything not all activity is what gets translated into protein. One could certainly imagine edits affecting mRNA stability, splicing patterns, translational ability, whatever the case may be. So we don't necessarily think all silent mutations, in fact, have zero effect on a gene. That being said, when we then look at the guide RNAs that either introduce a missense mutation in an essential gene or disrupt the splice site, or introduce a nonsense mutation, we see a much higher fraction of them now depleting the screen, suggesting that we are seeing good base editing activity as we would hope. We've done this across a couple different cell lines where and so the data from HAP1 are shown here, where we're just plotting AUC values, where we have a true positive rate if we're targeting non-sensor splice sites, and we're calling false positives here if it's either not introducing an edit or a silent edit. And we see different cell lines, all of them are much better than a random AUC value, which would be 0.5. Perhaps not surprising that HAP1, a haploid cell line, performs the best, but we do see good editing in some other diploid or higher copy number cancer cell lines as well. So this does seem to port pretty well across different cell lines. Very worth saying at the outset that, look, the false negative rate of this technology should absolutely not be ignored. If you can go back to this first slide, about half the guides that are predicted to introduce a nonsense or a splice site mutation don't score. So I would call those false negatives. But this is a screening technology. So we're looking to see what we can make statements about and not worry too much about what we can't. So one of the first screens we attempted to do with this technology in hand is ask questions about small molecule drug interactions. So here we're looking at the gene MCL1. This is a anti-apoptotic gene that is often overexpressed in cancers. So what we did is we made a library of guide RNAs that introduced every possible guide RNA along the length of MCL1. And then we introduced that library, and then treat it in 3 different conditions. So first, if we add nothing, since MCL1 on its own is not an essential gene, a loss of function mutation in MCL1, the cell should still be fine. However, if we now add a BCL2-like 1 inhibitor, because BCL2-like 1 and MCL1 have a synthetic lethal relationship, if you have a loss of function mutation of MCL1 and you've now inhibited BCL2-like 1 with a small molecule, then those cells should die. So we can use these 2 comparisons to find loss of function mutations in MCL1. But we can also find resistance mutations. If we treat the cells with both a BCL2-like 1 inhibitor and an MCL1 inhibitor, then if it's a loss of function mutation, MCL1, the cells are dead. But if the MCL1 editing, apologies, my screen keeps flipping forward. My daughter uses this computer for school too, so I'm just going to blame her for that. If we introduce a mutation in MCL1 such that it can no longer, that it retains its function, so MCL1 activity is still high, but it can no longer bind to the small molecule, then it will survive in those conditions. So what does that actually look like? So here we're comparing 2 of these conditions when we either treat with just the BCL2-like 1 inhibitor or the combined inhibitor. And we see, well, first, what we see quite nicely is that the intergenic controls are in gray, and they're nicely clustered in the middle there. But we do see some really strong outliers. So these are guide RNAs that either introduced a mutation such that the cells are now resistant to an MCL1 inhibitor, or they're sensitized. So there are MCL1 loss of functions, and on the lower left in the turquoise, or excuse me, the purple there, you can see an example is a mutation. So that's exactly what we'd expect. We introduce a nonsense mutation in MCL1; they're now sensitive to BCL2-like 1. But the resistant mutations are particularly interesting. So how do you go about validating these things? Well, it's real important to remember that this is very different from a CRISPR knockout or a CRISPR-I screen, where confidence in the screening result comes from using multiple guides per gene. So that's a good way to identify it. Of course, then one can validate it guide RNAs in a secondary screen. That's schematized on the bottom here. You can just tile across a gene and say, look, all these guides work. This is likely an on-target hit. With the base editor screen, though, the hit is almost always a single guide RNA because there are only so many PAMs across a gene. You're introducing an amino acid change at a specific position, so you can't just screen. The guide validation path is longer with these base editor screens. So how do we identify the causal mutation for these screens? So we now take our individual guide RNA, put it into cells, and then PCR. So now we're not going to sequence the guide. We're going to amplify the target site of the guide in the genome and look at how much that changes over the course of the screen by now sequencing the genomic again. This resistance guide as an example. Here's the locus where it targets in exon 1, right by an intron. So we can do this, add the guide, and then PCR. And we see that in the unedited condition, we see a couple of C to T mutations in the window where we expect. And then when we add the inhibitors, we see those mutations enriched. But we don't know which of those mutations is required for drug resistance versus which one is a passenger, just along for the ride, happens to be co-edited at the same time. Doing next generation sequencing and getting allele level information is really, really valuable. So what's shown here are the actual read counts from this sort of experiment. On the bottom is the wild type allele, though it's arrayed, and you can see, first you can see that the replicates look very similar to each other. So this is not an experiment that needs to be done many, many times to get good answers. So we can take the edits that we see and then translate into amino acid space. And you can see that in the left here, every allele that had an A227V mutation enriched. So that one mutation is both necessary. You can also see, for example, a T26M mutation came along for the ride sometimes, but is not on its own sufficient to cause resistance. We never see it on its own in the way that we do see the A227V mutation. And quite nicely, we see further down here, we see the A227V mutation not enriched, but that's because it's paired with a stop codon. So downstream introduced a stop codon, and that's why this allele was not functional, because it was not actually creating a full-length MCL1 with this A227V, but actually also introduced a stop mutation. By at the individual alleles that you actually make, that's a great way to distinguish causal and passenger mutations, which is going to be really important because our ability to predict exactly what edit is going to be introduced by a base editor. There's been some progress made, no doubt, but the predictions aren't perfect. So this validation step is really, really critical. This is sort of those same data, but now aggregated. If we don't add a small molecule, the editing efficiency of introducing this allele was about 30% or so. But now when we add in the molecules, all the alleles with this A227V mutation become a very substantial fraction of the population. Finally, quite nicely, we can then look at, well, where is this mutation? What is it doing? So this is CL1 bound to this inhibitor, and you can see that the A227 amino acid is right where the drug is binding. So it certainly makes a lot of sense that changing this amino acid would change the ability of to bind to its target. And in this case, it was very straightforward to then also make a knockout cell line that is exquisitely sensitive to an MCL1 inhibitor, and then do the rescue experiment where if we add back in this 7V mutation, that's the line shown in green, we see a substantial shift to the right. So they are far more resistant to an MCL1 inhibitor than when we overexpress the wild type. So a nice validation that what was true is also true with open reading frames. We've done the same thing for a couple of other drugs. Here's an example that I think is particularly nice. We're looking at PARP. So PARP inhibitors have had a lot of success in the clinic. Many different pharma have their own PARP inhibitor. 5 of them are listed here. So we did the exact same thing. We tiled across PARP, introducing all the mutations we could, and then screening to find resistance and sensitization mutations. And I think what's particularly interesting here, so this is this plot is a little convoluted, so I'll walk you through it. On the x-axis is the minimum z-score, so sort of how that guide performed the worst or the smallest magnitude of change, and on the for any of the PARP inhibitors. And on the y-axis is the maximum z-score across all PARP inhibitors. So what that gets down to is these that showed sensitivity to all 5 of the PARP inhibitors that we use, whereas up here are mutations that provided sensitivity to some PARP inhibitors for other PARP inhibitors. So an example of that is shown here, where this this validated G871K mutation, we see that in the presence of niraparib, basically niraparib has no effect on this mutation, but the other 4 PARP inhibitors, this edit very much to the PARP inhibitors. Whereas that exact same amino acid, glycine 871, if you change it to a lysine, you get this result. If you change it to aspartic acid, you get this result, where now all 5 PARP inhibitors are equally susceptible, or excuse me, all the variants of PARP are susceptible to all 5 PARP inhibitors. So you really get amino acid level resolution of how this small molecule is interacting with its target. Which so I would hope that as we develop more small molecules and really want to nail down the question of what is the target of this small molecule, something that obviously Tim just spoke about, and he mentioned Jason Shelter's work as well, that these experiments like this, I think, are going to be quite valuable. But of course, we're not only interested in small molecules. There are an awful lot of variants that are popping up in patient sequencing, of tumors, in somatic sequencing of just the variants that as humans we're all walking around with, and those are more or less collected in the ClinVar database. So as Ibrahim introduced, we certainly can't tackle all the variants that are in ClinVar with any one base editing technology. But there, about 13% of the variants that are in there can be introduced by 50,000 variants across about 3,500 genes. That's a totally screenable library. So we made that and we screened it. So this is a library that is intended to introduce every C to T mutation in any gene that's listed in ClinVar. Now I should point out that in ClinVar, there's a good number of variants that are listed as pathogenic, a relatively small number that are clinically annotated as benign, and a whole bunch that are just listed as variants of unknown significance. So some clinicians somewhere observed this variant in a patient and felt the need to put it in ClinVar. It's like, well, I've seen it, and I don't really know what to do with it. I don't know if this variant is breaking the gene, activating the gene, doing nothing to the gene. We just don't know. And I think solving that variant to function challenge is one of the next big frontiers in biology, especially bringing the promise of genomics to patients. So here we did a screen with this ClinVar library in 2 different cell lines, both non-haploid cell lines, Meljuso and HT29. We put in the library, and then we do a traditional dropout arm. We also push the system by either treating the cells with cisplatin to trigger DNA damage, or hygromycin to trigger translational stress. And we'll talk about the hygromycin. We didn't learn too much from that. We learned a little, but not much. But the cisplatin arm was really shown here. Each dot represents an individual guide. And what's shown is guide RNAs that deplete in the cisplatin arm relative to the TV no drug treatment arm. And you can see a handful consistently in the lower left. So these are guide RNAs that introduce some mutation that sensitize the cells to mutation. Now, when we look at what genes those guide RNAs were targeting, enrichment for guides targeting genes that are annotated as DNA repair genes. So overall in the library, only about, let's say 5 to 10% of the guide RNAs targeted a gene that is annotated as being a DNA repair gene. But then when we cut off the Z-score, so we're sort of looking in this region or in this region at different levels of threshold, we see a much higher fraction. So we're enriching for the guide RNAs that happen to target DNA repair genes. And here are just some of the top hits from both of the 2 arms, and there was pretty good consistency across the 2 arms. And you can certainly see that these are a lot of the frequent flyers or well-known genes involved in DNA repair. We can also then look at just the predicted effect of those mutations using a tool that the Ensemble website provides. And we see substantial enrichment for guide RNAs that are predicted to introduce a high impact mutation versus a low impact mutation, certainly suggesting that these results are making biological sense and these screens are working. Here's just another view of that now mapped at the gene level rather than the individual guide RNA level. And then when you look, when you put the genes that score into the string database, you see that there's a very high degree of connectivity, which is again quite expected in this case. So we think that these screens are working quite well to identify genes of interest. Now, of course, this is a bit of a tangent, but this was actually my favorite this whole study. So we had introduced these point mutations, but now we wanted to do a counter screen where we took that same library, but instead of pairing it with base editors, we paired it with wild type Cas9 to create loss of function mutations. We assume that most base edits that score are going to also be loss of function. It allows you to break a gene, but there could be some gain of function mutations. And in fact, I think we do find a little bit of evidence for that. So what's shown here again are each dot represents a guide RNA. On the y-axis is the performance in the base editor version, and on the x-axis with wild type Cas9, so the indel creating version. And so I'm particularly interested in the guide RNAs that fall into this category. These are the ones that when we presumably knock out the gene by introducing an indel, not much happens. But when we introduce a base edit, that base edit has a dominant negative. Just to give one example of a guide that scores in here, there's a predicted glycine to serine mutation in this gene TUB4A. This is a known pathogenic mutation in ClinVar. And basically what happens is when you introduce this change, now block the ability of TUB4A to partner up with all its other tubulin members and form filaments. Now, if you knock out TUB4A, there's a lot of other tubs that can substitute for it. So you don't see much of a phenotype. But if you are making TUB4A and you have this mutation in here, then you can certainly imagine how one bad TUB4A molecule can disrupt an entire filament being formed. So that's a nice example of finding a true gain of function mutation using this sort of screening approach. Now, from a couple slides ago, you might have seen that BRCA1 scored very strongly in the cisplatin screen that we did. So BRCA1, obviously a gene with very high clinical significance. We made another tiling library where now each guide represents a guide targeting the tiling across the window of BRCA1. And we see what we would introduce a defect or a nonsense mutation tend to deplete. Still see this challenge of false negatives. You can see some yellow dots up here in the non-scoring window. So I think especially when targeting genes of clinical interest, it's very important to not interpret these missense mutations as benign. All this means is that we have no idea did we introduce the missense mutation and it was benign, or did we just fail to introduce a mutation in the first place? So that's really important to keep in mind, again, especially for these clinically actionable genes. But we do see plenty of guide RNAs that score down here. I think it's interesting, and probably this is a trend that can most deleterious missense guide RNAs happen to be in this region highlighted in gray. That's the best annotated domain of BRCA1. We can then look at how well this screen distinguishes known pathogenic and known benign mutations according to their ClinVar annotation. We see AUC on that front. So BRCA1 seemed to work quite well. Now, of course, I'm sure everyone here is familiar with the work from Greg Finley and Jay Shanduri's group, where they tiled across BRCA1 using repair in HAP1 cells. And those data look extremely nice. But that is not a terribly high throughput assay. That took Greg, basically his entire PhD was dedicated to doing a lot of that. Whereas this was a screen that we could do in a couple weeks and get an answer. So if you can do it for BRCA1, why not do it for BRCA2? So we did. And you can see a similar story where, again, this is the best annotation. And this is where we certainly enrich for missense mutations. And again, when you do the AUC for separating clinical from, pathogenic from benign mutations, you definitely see that BRCA, this screen matches up very well with what's already annotated in ClinVar as well. So where are we going with base editors? First, I think it's, from this, we conclude that the CDT base editors can be used to conduct genetic screens in a pooled format. The validation path, I'd say, is very straightforward. It's just PCR and sequencing. But I would also emphasize that it is quite essential to really understand what is the allele that you actually made in your cells and how is that affecting the biology. I presented two types of screen, where you take all the guides you can, targeting across a region of interest. One use case for that, for sure, is looking at small molecule target interactions, for example, MCL1 or PARP1, but you can imagine doing this with all sorts of G protein interactions. Certainly genes of clinical interest, as I presented with BRCA1 and BRCA2, this variants of unknown significance or uncertain significance challenge is present now in the genomic age. So I think that screens like this will be helpful for helping us to understand at the amino acid level, what is a gene doing. I think it's really important to point out, obviously a lot of, you can do this with proteins, or even back in the day, you could mutagenize your cDNA in mutagenic E. coli and screen that way. And those are very helpful studies, no doubt. But here we're editing at the so you could also imagine doing this to map enhancer elements, promoter elements, splice enhancers, splice suppressors, UTR elements, et cetera, because it's all done on the endogenous allele. So I think that's going to be really, really helpful, especially for these regulatory elements. I've also shown that we can do this across. So one library we made was everything that we could make that's annotated in ClinVar. I think this will be really useful to start to understand hits that score in genome-wide associations when you merge base editing with some of the combinatorial screening approaches that we've seen folks talk about today. So we can look not just how two genes interact, but how two variants interact. And I think that'll be helpful. What GWAS is telling us, because I'd say right now, in many cases, the answer is we don't really know. So that'll be fun to figure out. So with that, I want to thank everyone in the Genetic Perturbation platform, the boat. I want to thank Ruth Hanna. She was an RA in the group who really led this project. She's now a graduate student at MIT across the street. I also want to thank JT Neal's group and the cancer program at Broad, and two of his RAs, Christian and Joseph, who were very helpful in this project as well. And with that, I am happy to take any questions. Thank you.
Thank you, John. I will start with one. So you mentioned that using base editors, obviously you can really cover every base in there. So how much more information do you think you will get if you add another base editor or use the CG base editor? Would that make a huge difference, or you think you already have enough information?
Yeah, that's a great on that. So, certainly, as Ibrahim mentioned, using Cas9 variants that have a broader range, so like Cas9NG or SPG Cas9, we started to just get a higher density because the PAM requirement is less, and those have worked very nicely. That said, I would encourage people not to use the XCas9 variant. That has not worked well in our hands. Likewise, the question is, well, what type of edit can you introduce? Here we use C to T, but obviously there's A to G, and even newer C to G. We haven't tried C to G yet, but we have started using A to G, or the ABE base editors. Version 7 of ABE didn't do much in our hands, but the ABE 8E variants certainly have looked promising. If you use just BE3, what density can you achieve across BRCA1? But then if you switch to the NG version, you can just tile way more guide RNAs and get a lot more information. Now, but for MCL1, you can see, hits are strong hits, especially are pretty sparse. But now with the NG, you could be pretty confident. Okay, like this is a region worth studying, this is a region worth studying, et cetera. So I think there's, certainly still a lot of tech dev to do, but mixing and matching these tools and putting them together, I think, is going to be absolutely a path forward.
Hi, John. Nice talk. I have two quick questions. So first is like for the knockout screen. So would there be any like a principle for design the guide RNA where you try to avoid like a repetitive C as a guide RNA candidate, or it doesn't matter? And second question is like for the claim the mutation from ClinVar, certainly you will limit to the guide RNA design region. So how do you see those bystander product? You will see it's like a negative factor, or like you could get some surprise benefit from those?
Yeah, that's a great question. So first, from the question of, your first question was basically about specificity. And yeah, I mean, just as with any other screening approach, you care a lot about specificity. So we would certainly not include a guide RNA in a library if that guide RNA was predicted to edit lots of places in the genome, because then it's really hard to figure out what's going on. So, including it off our line is absolutely still a thing one should do. As for the bystander mutations, are they a feature or are they a bug? That's a great question. Certainly when you get multi-mutations, it's nice to see, as I showed in one example, that you can really say, this is the allele or this is the mutation that caused it to change versus this one. So they at least in that case, effect on the cells. But you could also imagine for other cases, having more than one mutation could be useful. Just as one example, let's say you're studying phosphocytes, and let's say you can't mutate the serine that is known to be phosphorylated, but let's say you could mutate, to the right. That might disrupt the kinase binding domain that is phosphorylating it in the first place. So in the short run, there's not much you can do about them. So you just sort of need to take what comes and temper your expectations appropriately, and of course know that there might be bystander mutations and analyze your screen appropriately.
So what would you do if you have, like, your target gene has homologous sequences in the genome, some like nearly identical?
Yeah, well, I mean, I think Trevor's talk gave a great introduction to why, if you were only to look at one of the genes, if they're very highly homologous, you might erroneously conclude that, oh, this gene doesn't do anything, because of course the paralog is compensating for it. So if it were the case that you could introduce essentially the same edit across both paralogs, that'd be great. My guess is in most cases that won't be true. I mean, paralogs are highly homologous, or can be highly homologous at the amino acid level, but certainly at the wobble positions, they're pretty free to drift. So generally, I don't think that'll occur too often, but certainly one could be opportunistic in some cases and really leverage that to one's advantage.
Here's one question: to lose the PAM constraint, would you recommend, I guess, recently reported PAM? Would you comment on recently reported PAM-less Cas9 to be used for base editing screening?
Yeah. So this is a reference to the SPRY variant of Cas9 developed by Ben Kleinsteiver and colleagues. Yeah, we're actively working with Ben to figure out how well those work in base editing applications. Certainly, if you didn't already going down to the NG variant, where the PAM is more or less just a G, that really increases your density, but not even needing to worry about that and using a truly PAM-less Cas9. Stay tuned, but I'm excited to.
Oh, thank you again, John.
Yeah, thank you, everyone.
Last talk, our last speaker is Andrew Anzalone. He was a postdoc in David Liu's lab, where he developed this exciting PAM editing, and now he's the head of Prime editing platform at Prime Medicine. I think we will have a lot of questions for you, Andrew.
Okay, great. Thank you so much for the invitation to speak today. Thanks for putting everything together, all the organizers. So today I'm going to talk about prime editing. So this is an approach that I developed while a postdoc in the Liu laboratory, and it's a method to do genome editing in a pretty precise and versatile way that doesn't require any double-stranded breaks to be generated or any donor DNA repair templates. So I'm sort of speaking to you from my recent new post at Prime Medicine, where I'm working to develop this technology for therapeutic applications, but all the work that I'll present to you today is from the Liu lab at the Broad Institute. So I'd just like to open up things with sort of giving everybody an idea of what types of mutations cause human disease, in particular human genetic diseases. So on the left is a pie chart showing the different types of mutations that are found in the ClinVar database that are annotated as pathogenic. And we can see that about 50% of those mutations are point mutations, broken down with about 60% of those point mutations being transitions, so C to T or A to G, and the remaining being the different transversion point mutations. There's also a pretty large portion of the pie that's made up of deletions and duplications and insertions and deletions. These are also very important for various diseases. So on the right, I'm just showing you different examples of diseases that many of us have heard of, like sickle cell disease or cystic fibrosis, that are caused by these different variants. So to just speak to a little bit of how, CRISPR technologies can be used to address these mutations, either for basic research or for genome editing, we're all very familiar with Cas9 nuclease. And this enzyme uses a single guide RNA to identify its target site in the genome. And it has a restriction that it requires a PAM adjacent to the target site. And it also has two separate nuclease domains, one a REV-C domain that nicks the PAM containing strand, and a H&H nuclease domain that will target and nick the target strand that's bound to the single guide RNA. And using the Cas9 nuclease, you can generate double-strand breaks very robustly at different places in the genome. And these double-strand breaks are repaired through one of two main pathways, either an end-joining pathway, where in the absence of any type of donor DNA, generally will produce a wide mixture of indels that can be used to effectively disrupt genes. Alternatively, if a piece of DNA is provided in addition to that, DNA, double-stranded DNA, can be inserted at the site of the break through end-joining mechanism. So this is a mechanism to integrate some exogenous DNA into a genomic locus. Alternatively, a separate repair pathway, one that's much more precise, called homology-directed repair, can be used. And either single or double-stranded DNA templates can template the repair of that break and lead to very precise mutations like point mutations or small insertions or deletions, as well as the integration of larger pieces of DNA. So this is obviously a very appealing strategy for doing genome editing and has been used widely in research. However, when it comes down to it, when you look at a lot of cell types that are relevant for therapeutics, it turns out that nucleases that generate double-strand breaks aren't very well suited for doing these precise corrections because the homologous repair pathways tend to be not active in post-mitotic cells. And there are also additional complications with delivery of the donor DNA repair templates. So this really served as motivation in the field to develop other genome editing technologies that can make edits without relying on these particular repair pathways. So you've all already heard a lot about base editing today, so I won't go through it in very much detail. The Lulu lab previously developed two versions of base editors, a C base editor and an A base editor. And as I mentioned on the previous slide, this is representing about 30% of all the pathogenic mutations annotated in ClinVar as transition mutations. And in addition to that, recently C to G base editors have been developed that account for a portion of the transversion point mutations. Even though these tools are very powerful and have lots of applications on their own, there's still the remainder of this pie that is really yet to be addressed in other ways beyond base editing. And in addition to that, even for the transition mutations and C to G mutations, there's not always a correct PAM, appropriately suited PAM or amenable target site for using base editors for these transitions. So even for this category, there is a reason to look at other technologies for performing these edits. So I'll tell you about the technology that I developed in the Lulu lab that we call prime editing. And this is sort of conceptually a search and replace mechanism for doing genome editing. And again, without requiring a double strand break to be generated or requiring any donor DNA template. And so there are three components to a prime editing system. One is the Cas9 domain, and we use a nickase, so that does not generate a double strand break and instead only nicks one of the two strands of DNA. This nickase is fused to a reverse transcriptase enzyme, and that's the enzyme that will be responsible for actually writing in the edited piece of DNA into the genome. And lastly is a pegRNA, which is a single guide RNA that's been modified such that its 3' end has an extended sequence that serves as the template for DNA repair. And it's a reverse transcriptase enzyme that's going to read that template and write the edited DNA sequence directly into the genome. So on the right side here is sort of a schematic of what types of edits you can do with prime editing. So everything starts sort of with the targeted sequence in the protospacer. And in this particular example, we're using SPCas9 sort of as the nuclease. And the pegRNA induced nick site is shown by the purple arrow, and anything 3' of that position of the nick, in principle, can be edited by a prime editing system. From that position onward through the PAM, is where you can synthesize new pieces of DNA that will replace the existing DNA sequence. And I'll try to show you throughout the talk that prime editing can do all types of point mutations, whether they be transition point mutations or transversions. We can perform insertions of up to and over 40 nucleotides, and deletions in the range of around 80 base pairs have been accessible. And then various combinations of those types of mutations. And really, in principle, it carries a lot of the versatility that homologous repair carries for doing small, precise changes, because the edit is encoded by a nucleic acid template. So I just want to walk through the steps of prime editing quickly. So just to reiterate, the prime editor protein is this fusion protein of Cas9 nickase to a MMLV reverse transcriptase enzyme. And then the pegRNA is used as both the targeting sequence, and so the spacer specifies which position the genome will be edited, and then also this extension carries the editing template. So the first step is the Cas9 complex will bind and recognize its target DNA sequence. The RUBC domain of the Cas9 nuclease, which is the remaining active nuclease domain, nicks this non-target DNA strand. So this is the same strand that contains the PAM sequence and forms an R loop upon Cas9 binding. So once that strand has been nicked, it was known from biochemical experiments from Jacob Korn's lab in particular that strand is quite flexible and can actually hybridize to other nucleic acids. So we reasoned that after nicking, this DNA strand could complex with an extension on the RNA to form sort of an RNA DNA duplex and serve as a priming site for the initiation of reverse transcription. So as you can see in the left schematic, we now have a genomic DNA primer hybridized to a complementary primer binding site in the pegRNA, and that system is now perfectly poised to undergo reverse transcription by the associated reverse transcriptase enzyme. So that reverse transcriptase will copy in new edited DNA sequence that you've programmed directly onto that piece of genomic DNA. And so this step only gets us so far. At this point, what we would imagine occurs is that we have a new 3' flap sequence that contains this edited DNA strand. And as we were considering designing this editing strategy, we considered how this might be repaired. And one thing that we imagined is that if there is some homologous sequence in this flap downstream of the edit sequence, shown in red, it might be possible that that 3' flap strand could invade the adjacent DNA and displace a corresponding 5' flap sequence that contains the original DNA sequence. And then we had reason to think that that 5' flap could be preferentially removed because it resembles certain intermediates of lagging strand DNA synthesis, as well as base extension repair intermediates, where these 5' flap sequences, in this particular example, part of Okazaki fragments, are efficiently excised, and then the resulting nick is ligated to complete that fragment of DNA synthesis. So based on this resemblance, we thought it might be possible that certain structure-specific endonucleases like FEN1 might be able to remove that 5' flap and close that strand. And so if that were to occur, you would have this heteroduplex of an edited strand and an unedited strand, and then further DNA repair through mismatch repair or replication across the edited strand could lead to an edited DNA sequence. And the hope was that even if this weren't successful, repeated attempts could potentially drive the process towards edited DNA, as long as a lot of byproducts that were not generated in the process. So we did many biochemical experiments and other experiments leading up to our work in mammalian cells, but I'll skip those in the interest of time and just jump ahead and show you sort of what ended up being the starting point for our mammalian cell genome editing work. So on the left here, I'm showing a diagram of a pegRNA. And for the most part, if you start at the 5' sequence and move forward, it contains the same elements as a single guide RNA. So it contains the spacer sequence and the scaffold of the guide RNA. But then once you reach the 3' end of the canonical scaffold, this is where we've appended a repair template or within this 3' extension sequence. And this repair template contains two really important elements: the actual template for DNA synthesis that the reverse transcriptase will copy, and that includes the edited sequence, as well as some sequence that is very homologous to the site, so that it facilitates DNA repair. And then there's also the primer binding site downstream of that, which again serves as this hybridization point for initiation of reverse transcription. So in red here is the base that we're targeting to edit, corresponding to the DNA sequence shown at the top right of the slide. And the idea is that Cas9 in the prime editor will nick the DNA strand, and that green sequence will serve as the primer, hybridize to the primer binding site, synthesize across that template, and then replace this T position with an A nucleotide. So in our very early examination of this, we found out some pretty simple things. In comparison to in vitro studies that we had done, we found that the primer binding site in cells needed to be much longer to support any editing. So where in vitro we could get away with 5 or 6 nucleotides in a primer binding site, we found that we needed to extend that sequence much longer. In this particular case, 13 nucleotides being optimal in order to see some editing activity. And we also found that the fusion of the reverse transcriptase to the C terminus of Cas9 as opposed to the N terminus was superior. So we call this first generation of editor PE1, which contains the Cas9 nickase fused to the MMLV reverse transcriptase at the C terminus of Cas9. And so this was a very exciting first demonstration of editing, though the editing efficiency here is quite low. And so given that the reverse transcriptase enzyme from marine leukemia virus has been extensively engineered for different applications, we were very eager to explore these mutants and see if they improve prime editing activity. So we took various mutations that had been reported in the literature and examined them individually or in combination with other mutations. So we looked at around 19 total variants in total and examined these across various editing loci. And eventually we arrived at this PE2 prime editor variant that contains the Cas9 nickase fused to an MMLV enzyme that contains 5 mutations. And so that PE2 editor we found was much more efficient at prime editing by comparison to PE1. So the other component of the prime editing system that we sought to optimize and also just examine and characterize was the pegRNA. And so as I showed on the previous slide, we've already noted that the primer binding site length of a pegRNA is quite an important factor in how efficient the prime editing system will be. So on the top of this slide, I'm showing 3 different loci and profiling the primer binding site length and editing efficiency at those sites. And we can see that there are certainly optimal lengths for supporting efficient prime editing. And also another thing to note is that there's not a single length of primer binding site that you can say would be optimal across many different sites. So the profile varies quite a bit among these different edits. What we also found early on was that the prime editing system can install insertions and deletions on the smaller side. So single insertions or deletions and a 3 nucleotide insertion, and PE2 does this more efficiently than PE1 as well. So the other component of the pegRNA extension is the reverse transcription template. So this is the sequence of DNA that will actually be synthesized. And we also found that the length of that sequence can be an important determinant of prime editing efficiency. So again, we profiled across 4 different sites various lengths of RT templates. And what you can see from these, I think is somewhat apparent, is that many different RT template lengths will support efficient prime editing. However, there is not one, you wouldn't be safe choosing just a single length without knowing any information, because it's very likely you could end up on one with particularly low editing efficiencies. So overall, we can see from this data that the PE2 enzyme is much more efficient than the PE1. It's actually tolerant of suboptimal primer binding sites much more than PE1 is. And we can see an increase in efficiency of between 2 and 5-fold, depending on the pegRNA design. And in addition to that, there are many RT templates that support efficient prime editing, but some of them, this sort of should be a sampled screen to see which pegRNA design works most efficiently for a given site. So these are some of the early rules or guidelines we learned from our study. So the last way that we approached improving the editing efficiency was to try to bias the DNA repair system to incorporate our edit. So I'd like to just walk sort of stepwise through the prime editing process the way that we view it occurring. And the first step is that you're starting with this DNA sequence where you'd like to replace the positions labeled with the red X with a new sequence labeled with the green Y. And so in the prime editing mechanism, we suppose that a new 3' DNA flap is synthesized containing the edited DNA sequence. What we hope doesn't happen is that the 3' flap is simply excised, the DNA is religated, and you go back to the original DNA sequence. So while we hope that that doesn't happen, in some ways, I think in a lot of cases at least, this will be okay because the substrate remains unchanged, which is to be distinguished from if a double-strand break is generated, wherein in many cases, this will lead to disruption of the site by the formation of indels and no longer will be a substrate for editing. So if this sequence at least remains unchanged, it can be serving as a substrate for subsequent rounds of editing attempts. But what we do hope happens instead is that this 3' flap can invade the adjacent DNA double helix and release the 5' flap sequence. That 5' flap can be excised and the resulting nick ligated. And at this point, we have a heteroduplex of DNA where one strand is edited and the other still is unedited. And we want to drive DNA repair to replace that unedited strand with a new edited strand. And to do that, we thought we could take a trick sort of from the base editing playbook, which is to nick the non-edited strand and thereby try to trick DNA repair to fix and replace that strand. We call this strategy PE3 or PE3B, depending on slight differences in the strategy. And what the system is it delivers a second guide RNA, and this time just a simple single guide RNA that will target the opposite strand and nick that strand to direct the DNA repair machinery to replace it. And then hopefully this preferentially results in the repair to incorporate the desired prime editing outcome. So this strategy we found works quite effectively across a number of sites. Conceptually what we're doing is identifying a position or a sequence on the bottom strand here in this diagram where we can place a single stranded nick and favor replacement of the non-prime edited strand. So when we provide these additional single guide RNAs, we can see somewhere between 2- and 4-fold increases in prime editing activity, albeit it does come with associated increases in indels in particular cases. So what we have found in general is that nicks that are downstream of the prime editing position, so on the plus annotation in this notation, those nicks provide higher editing efficiencies and fewer indels than if we nick on the minus side of that strand. And in addition to that, the distance of the nick induced by the single guide RNA relative to the position of the nick induced by the PEG RNA is important. So if the nicks are too closely spaced at particular sites, that can lead to higher indels. So this is another variable that requires some screening in order to find a compatible single guide RNA sequence to promote high efficiency PE3 editing. So one of the concerns, of course, with this approach is that we're generating higher levels of indels with PE3. And so one way we thought of getting around this is to make that second nick occur after the prime edited strand is repaired. So what we hypothesize is that the double strand breaks generated from nicking both strands simultaneously are what lead to the indels that we observe in the PE3 system. So if we could temporarily separate those two nicks, those two nicking events, such that they do not occur simultaneously, we would propose that that would lead to lower levels of indels overall. And the strategy to do that was to design a single guide RNA spacer sequence that actually contains the edit so that this spacer is less likely to bind before the edited sequence goes in. And also it's likely to be overlapping with the PEG RNA spacer such that two Cas9 complexes could not bind simultaneously in any event. So the hope is that after we get to this intermediate of a heteroduplex where one strand is edited and the other is not, we can make a new Cas9 nickase recognize the edited DNA strand, nick the opposite strand, and promote repair to incorporate the edit. So we found that this strategy can be quite effective when it is an option. As we can see here at this RNF2 locus, a prime editor PE2 system where the second nick is not incorporated leads to roughly 10% editing. That editing efficiency can be increased to about 30% if a single guide RNA nicking downstream 41 base pairs from the pegRNA nick site is included. However, that comes at the cost of about 5 to 8% indels. However, if we use this PE3B strategy where the nicking guide RNA recognizes the edited strand specifically, you can see that the editing remains high and actually increases. However, the indels decrease substantially. This was true for two particular edits at this locus. And I think a pretty striking example is this last edit, this +7A to C at FANCF, where we see almost equivalent levels of indels and correct edits when we use the traditional PE3 strategy, but moving to PE3B almost entirely eliminates the indels and maintains the improved editing efficiency that we see with PE3 over PE2. So we recommend that when PE3B is an option, that it be explored. However, it does require nested PAM sequences and that second PAM for the single guide RNA, so it's not always an option. So we were able to show that prime editing is remarkably versatile in the types of edits it can install. So we can incorporate, again, any point mutation. So this is the top left graph is showing an example of every single possible nucleotide change within 8 base pairs of the nick site at a particular locus. And this is something that we can do at many different sites. We can also do deletions up to 80 nucleotides with prime editing. As smaller insertions and deletions across various sites, and combination edits where we can do a 3 nucleotide insertion and a deletion somewhere else, or we can do a point mutation at one position and an insertion slightly downstream. So sort of any combination that you'd like of these different changes. And one other really favorable feature that we found is that prime editing can potentially be used to make edits very far away from the pegRNA nick site. And this reduces the sort of dependence on having PAMs very close by to your target nucleotide. And as a result, prime editing is less constrained. So we're able to see that we can install edits 30 plus nucleotides away from the nick site. So this is a really, I think, attractive feature of prime editing. And we found that this is possible in many other cases as well. It does increase the complexity of the pegRNA because the further away that edit is from the nick site, the longer the template must be in the pegRNA. But I think we found that in many cases, this is still possible. I just want to show that the prime editing does work in other cell types. So all the previous data was in HEC293 T cells. This is showing prime editing data in some other cell lines, including K562, U2OS, and HeLa. And excitingly, even though editing efficiency was on the lower side, we did see prime editing in post-mitotic mouse primary neurons. So this was promising because it sort of implies that prime editing does not require DNA repair dividing cells and cell replication in order to function, which is the case for a lot of homologous repair processes. In addition, prime editing, I think, compares favorably in many different cell types to Cas9 initiated HDR. And when we looked across these different cell lines and compared the two editing processes, we found that prime editing would either be a higher editing efficiency for the desired edit or a similar editing efficiency as Cas9 initiated HDR. But in all cases across the board, the byproducts, in particular the indels, were far lower in the prime editing systems. So the edit to indel ratios were much higher for prime editing. And so we were able to use prime editing to install various and correct various pathogenic mutations. So one of those being the A to T mutation that's responsible for sickle cell disease. So we were able to both install this mutation into HEC293 T cells, isolate those cell lines, and then correct that same mutation just by changing one nucleotide in that pegRNA sequence. Likewise, we were able to insert the 4 nucleotide sequence that is responsible for a large number of Tay-Sachs alleles. and then similarly correct that mutation. And also install a G to T transversion mutation in the prion protein gene that confers resistance to prion disease. So all of these edits we were able to achieve with fairly high editing efficiency and a very favorable edit to indel ratio. And the last thing that I wanted to show was sort of the off-target activity of prime editors and how it compares to sort of Cas9 off-target sites. So this first panel of data is showing the off-target activity of Cas9 nuclease at 4 previously characterized sites. HEC3, HEC4, EMX1, and FANCF. The top row of data is showing the efficiency of on-target editing with either single guide RNAs or various PEG RNAs that we had designed. And we can see that editing activity is high, fairly high across the board. And then in addition, 4 off-target, known off-target sites for each of these 4 loci. And we can see that there is substantial off-target activity at several of these sites. In particular, an off-target site for HEC4 that is much higher editing efficiency than the on-target site. So this is a sort of previously established profile of Cas9 off-target editing. And when we do the same comparison to prime editing, we see that the on-target activity that we have at these sites is modest to high. But the off-target editing is incredibly low or undetectable at many of these sites, with the exception of this off-target site 3 at HEC4, which is that same site that produces more indels than the on-target site. And it turns out when you actually go in and look at this loci, it's very, very similar to the on-target locus, and there's not a good reason to think that there wouldn't be efficient editing at this site as well. But it does depend on the PEG RNA that's used, and it turns out that depending on how well the homology in the PEG RNA matches the locus, changes whether or not editing is efficient at this off-target. And since our original publication, there have been several other papers published looking at off-target activity, and in general, these have given prime editing a favorable report for having low off-targets. So I'd just like to summarize, but hopefully I've communicated to you that prime editing is a strategy to directly copy edit information into genomes without creating a double-strand break or requiring donor DNA. We generated 3 different systems of prime editors, going from prime editor 1 to prime editor 2, and finally prime editor 3. And these each offer successfully higher editing efficiencies. And we can get sort of reliably between 20 and 50% editing in HEC cells for a given target site without any bystander edits, meaning that we get fairly precise editing of the target nucleotide, fairly low indels, and low off-targeting activity compared to Cas9 nuclease. And we've been able to show that prime editing can occur close to the PAM or far from the PAM sequence, and can perform all types of mutations, so transitions, transversions, insertions, or deletions, or a combination thereof. And we think that in principle, this can be used to correct many different pathogenic alleles that are associated with disease. So with that, I'd just like to thank and acknowledge everybody in the Liu Lab, in particular, Professor David Liu, who's been an excellent mentor and established an amazing research environment at the Broad Institute in his laboratory. And I'd also like to thank everybody who worked with me on the project listed here on the left. And in particular, I want to call out 3 people from the Liu Lab that have sort of joined me on this new, embarking on this new mission to translate prime editing into therapeutics at Prime Medicine. So John Levy, Chris Wilson, and Ariel, are all with me at Prime Medicine right now, working on prime editing and using it for therapeutic applications. So with that, I'm happy to answer any questions.
Thanks, Andrew. Do you envision one day prime editors will just completely replace base editors?
That's a good question. I don't think that's likely to be the case, and there are a few reasons for that. The first being that the prime editing window and the base editing window are actually quite distinct. So base editors edit nucleotides very early in the spacer sequence. So in the position sort of between 2 and 10 of the protospacer, whereas prime editing edits from position 18 in the protospacer and 3' of that into the PAM and downstream sequence. So that non-overlapping window, I imagine, based on which spacer sequences work well and other factors, would determine whether, just alone would determine whether prime editing or base editing is a better strategy. And at the current time, I think that base editing has a higher average editing efficiency than prime editing. So, you know, depending on how that changes and evolves over time, base editors could be the better choice in a lot of cases. However, in other cases, base editors will have bystander target nucleotides that can't be edited. And for example, if two consecutive A's exist in the window of the base editor, that's where prime editors would have a distinct advantage of targeting just a single one of those nucleotides. So I'm hopeful that both technologies sort of develop in the future and become even more efficient and applicable. And I foresee that there will always be a time where both have their uses and they have complementary strengths and weaknesses.
Could you comment on the non-specific RT activity in the cells?
Sure. So we haven't observed any non-specific activity up to this time, in the sense that the reverse transcriptase is reverse transcribing cellular RNAs and leading to a change in the abundance of those levels. So we performed RNA-seq experiments of cells transfected with prime editors or transfected with just Cas9 nickase, or transfected with prime editors where the reverse transcriptase domain is inactivated. And in general, we did not see significant changes in the transcriptome between those cells, except that those transfected with the Cas9 nickase actually induced a heat shock response. So probably because the expression was much better for that protein. But other than that, we didn't notice any areas where the transcriptome was significantly changed.
Here's one question asked about what is the ability for prime editors to multiplex?
Yeah, that's a good question. I think it's certainly possible. We've done that in the past. You know, if you provide 2 pegRNAs to a cell, we generally can observe edits at both positions and bulk sequences. We haven't gone to look at how many cells contain both edits. So we haven't, you know, we haven't grown out single cell clones or done any type of single cell sequencing that would let us get at that question. But based on the efficiencies of those edits, we think it's very likely that that's possible. And so the question becomes, if the editing efficiency of each site is only 20 to 30%, maybe only 5 to 10% of cells would contain both edits if they were not dependent at all on one another. So, so we do believe it's possible, but we haven't really investigated thoroughly yet.
Do you think the binding of the primer, the primer binding site actually confers additional base pairing that increases specificity?
Yeah, that's a good question. So this is definitely something that we, even in anticipation of developing the system, we thought about and we're concerned about. So by virtue of the way everything is designed, the primer binding site of the pegRNA is complementary to the spacer sequence of the pegRNA. And that's because it has to be complementary to the nicked protospacer sequence. So as a result, if you did an RNA secondary structure prediction of a pegRNA, what you would likely find is that the primer binding site makes a hairpin with the guide RNA spacer. And this could affect for sure the targeting activity of that pegRNA. And in the off-target work, we do see indeed that off targets tend to be lower with Cas9 nuclease using pegRNAs compared to single guide RNAs. So we do think it influences the activity, and it's an area of potential improvement in prime editing systems.
Okay. So here's a question regarding the longer pegRNA being longer, and this is asking about whether the secondary structure, well, the unintended secondary structure because of the longer RNA would affect the prime editor activity. I guess it's probably because they had similar experiences. We were really excited about prime editors, of course. So we were able to reproduce the activity using your published prime editors, but when we design our own, we just don't know how many different combinations of primer binding site and RT template to use to find good pegRNAs. I think we might have a couple active ones, but most of our own designs are not working. So I think this question is probably related to that.
Yeah, I can speak to that generally. So yes, as you increase the length of the template, you're increasing the probability that some secondary structures will form in that pegRNA. In some ways, that could be a good thing, that the extension is structured. That might make it more or less susceptible to degradation. Obviously, you wouldn't want it to be structured in the wrong places such that it prevents reverse transcription. However, we've inserted sequences that are predicted to form very strong hairpins, like LOXP sequences, which would predict to have a 13 base pair hairpin formed. So it doesn't seem that that structured sequence within the template is problematic for the reverse transcriptase. So that being said, again, if the extension sequence is forming RNA secondary structures that either disrupt the guide RNA scaffold, or block the primer binding site, or even just make it more susceptible to degradation, that could be problematic. And so the stability of the pegRNA is a big question, and it's one that hasn't been addressed yet to my knowledge. So we don't know if that extension is resected in a lot of cases, or if there are different variables that affect the efficiency of that resection. So that's still a very open question, and I think a very good one. And then lastly, to the point of the pegRNA design, what we have tend to do is screen a small series of pegRNAs in our initial pilot experiments. So you might pick 4 primer binding site lengths and 4 RT template lengths and make a matrix of 16 different pegRNA designs and test each of them. What you're likely to find is that one of them is significantly better than many of the others, just because there tends to be a peak for each of the 2 dimensions. We don't know as much of whether or not the specific edit affects those parameters. So if you were to use that same pegRNA to try to change the mutation, they might no longer be optimal. So we haven't characterized that. That was sort of our working assumption. So a lot of the experiments that are, a lot of the data in the paper use a fixed primer binding site length and template length for a given target site. Though we don't know if that carries in all cases to once you change the edit that you're trying to install. So our sort of recommendation is to do a small pilot screen, and preferably in a place like hex cells where it's easy to look at many different variants just by cloning the plasmids and transfecting plasmids.
Have you tried other RTs? Would that make a difference?
We have, and you know, the preliminary work that we did very early on, we looked at a few others, and nothing was anywhere close at the time to the reverse transcriptase from marine leukemia virus. Since then, others have looked at it. So there's a paper that was published, prime editing in plants, that used a cauliflower mosaic virus reverse transcriptase, as well as a bacterial retron reverse transcriptase. And both of those supported prime editing efficiency in plant cells. So I think there are other options for the reverse transcriptase enzyme, and you could think of various reasons why you would want to explore that, whether it be reverse transcriptase activity, protein expression, or even interaction with other cellular components. So that's a really interesting area to explore, I think.
All right, thank you. So while Andrew gives the last talk today, we are not done yet. We have a panel discussion after now, and Yishen Chen, who's an assistant director at the GEIC in charge of iPSC projects. He will give a very short presentation on preliminary data using base editors in iPSCs to start the discussion, and Trevor will co-lead the discussion. In addition to the speakers, we also have Steve Hoffman from Illumina joining us with the discussion. Yishen?
Okay, thank you so much. So I think Ibrahim has given a nice introduction for base editor, and today just give a quick tech note for use the base editor for cell line generation. So we can see here this pie chart showing the different type of human pathogenic mutation from ClinVar database. As you can see here, over 50% of those genetic variant is a single point mutation. And among those single point mutation, you can see nearly 50% of those mutation could be reversed by A to G change. And genome editing offers a powerful tool to study those pathogenic variant by either introduce the mutation or like a reverse the mutation in desired desired model system. And CRISPR-Cas9 system has been the predominant way for conduct genome engineering. And so after the DNA double-strand break, there are different scenarios. You could first introduce a premature stop codon, which will result in genetic knockout genotype. And or you can provide with the single strand or double strand donor template, use the homologous direct repair system to introduce mutation or insertions. And also the deletion could be done with the pair of a guiRNA approach. So here is the case, here's the project we have done before. So the goal here is to introduce the G2A change, and the guiRNA we use is upstream, a little bit upstream of this mutation. And the goal is to introduce heterozygous mutation for this G2A change in a wild type iPSC cell line. And with the standard Cas9 and double-strand DNA break, we certainly can achieve really high editing efficiency if we combine the modification with block or the silence mutation only. We can achieve almost like 20% through the HDR pathway. However, we can also see the other small indel happen, which combined has like 40% in the total population. So if we look the single cell data, if we perform the sequence alignment from the two independent clone, we can see here, although we have the maybe one allele has a silent mutation and or the desired mutation and silent mutation in one allele. However, the other allele also has like unintended indel, like +1 or -1 indel in the other allele. So the editing outcome is less ideal. So what could we do better to achieve a better editing outcome? Because in this scenario, we often need to retarget or like screen more clone. So that brings us to use the best editor approach. So one of the nice, great advantage for best editor is like best editor doesn't require a double-strand DNA break to edit the target site. And there are two main flavors for best editor, like Abraham has talked about, like CBE or editing ABE best editor. And they are like a C2G best editor as well. So they are different flavors. However, best editor is not of the limitation. There are certain limitations for use the best editor to do the editing. So for example, so the target window is very small. You only have very limited active window, and or you could what could happen is like you might have the bystander byproduct in the outcome, and or it could possible like you might have your intended mutant site, target site is far away from activity window, or like the sequence content nearby your desired mutation site is not ideal, so lead to like inefficient editing outcome. So to overcome those limitations, a variety of best editor has been developed over the year, and which allow the best editor to be more versatile in different scenarios. So here is a case collaboration project we are doing with David Liu's lab, and then also an investigator at WashU. So the purpose is to, this is a patient-derived iPSC line, and so the patient harbor a mutation which is C2T mutation, and result in a premature stop codon downstream. And you can see it was a G2A change in the reverse strand. So the purpose to use the base editor is like we use the ABE base editor to reverse this A back to the G. And because the pen size NG, so we use the SPCas9 NG variant for this editing. And we test two different formats, two different ABE version and two different format of the delivery method, protein and RNA. So if we can see here, we can pick up nice activity within like 48 hours. So we can reach maybe 30%, over 30%. And over as the time go along, we can see like a higher editing efficiency could be achieved with the mRNA format of base editor. And if we separate protein and RNA, we can see the protein kind of saturate after 72 hours, and we can achieve like around 30% efficiency. However, with the modified RNA, let's increase the stability, we could achieve almost like 80% editing efficiency. And if we look the outcome from the single cell clone, these are the 6 clone we only need to screen a handful of clone because editing efficiency is so high. So from this 6 clone, we can identify clean homozygous correct clone, and also nicely we also have the heterozygous clone, nice clean heterozygous clone as well. So I think the best editor, the Nobel Prize for CRISPR has set a great stage. And best editor, I think recent year just also reached several milestones. For example, recently there are more versatile version of best editor, like you can do like simultaneous CDT, ATG mutation, which is very, could be used for the saturation mutagenesis analysis or like a library screen. And there's a new shelf in town, right? Like the Angel present the prime editor. And we also have the poster section by our scientist, Z1, to demonstrate the real user experience. So please check out the poster section. And so combined with the talent approach, so CRISPR is the first time has achieved, be able to edit in mitochondria genome as well. And recently, with the AAV delivery method and split Cas9 approach, you can actually, show like great therapeutic potential with the best editor. So to end the talk, I would like to thank the group from GIC, and this is our, the photo credit is from Xiaoxia, and also Greg from David Du's lab, and then Jordana and Anton from TriLink Biotechnology for provide the reagent. So I think if people has further question for my talk, you can submit to the Q&A section, and I think we can save time for the panel discussion. So continue. So now we can start the panel discussion if people don't mind. So I think today we still have most of the speaker within our panel, and to continue the conversation with the best editor. So I think the first question I have for the panel is like for the best editor, how do you see like those CRISPR independent off target, and how to do the off target with the best editor, which is quite different from the traditional CRISPR-Cas, which introduced the double-strand DNA break. So I think we can start with Shenda. I would like to hear your opinion on that, and maybe John and Ibrahim afterward. Thank you.
So your question is, how would you understand the genome-wide activity or off-target activity of base editors, essentially, rather than nucleases, right?
Yes, yes. Mm-hmm.
Yeah, so that's a good question. I think, of course, a lot of the initial work has been focused on the nuclease genome-wide activity. I think that there's a number of ways that we and other people have started to look at the genome-wide activity of base editors. I think one of them has been, in addition to like in silico approaches, to using methods like circle-seq and change-seq. So looking at positions where, for example, Cas9, which a lot of the base editors are based on, where they might actually make double-stranded breaks, and then just analyzing them for additionally, whether or not there might be base edits. So that's kind of one starting point, and I think has been, somewhat successful in finding the subset of nuclease sites that can also be base editing off-target sites. So you kind of imagine they might be shared. Other people have also used methods like digenome-seq, which is a whole genome sequencing-based approach, coupled with enzymes that can convert deaminated sites to double-stranded breaks. So this could be like user enzyme or endo5 to basically now try to sequence for areas that have base editing activity. We have also started thinking about, I've been working on adapting methods like circle-seq and change-seq directly for base editors. So this is again similar to the digenome-seq method, where with base editors, typically you have a nick and a deamination event. You're converting the deamination event to another break so that you can then specifically sequence for those areas that have been modified for base editors. So I think like that summarizes, I think, the number of different approaches that are out there. It's not quite as mature or advanced as the methods for nucleases, but there are a number of different, mostly biochemical methods for figuring out where there's base editing activity, and then you can confirm those with targeted sequencing.
Yeah, so I completely agree with what Shengdar said. I think another version of off-targets that we should consider when using base editors, part of this is we're overexpressing an APOBEC domain in cells. And I know from doing CRISPR-A screens, one of the most lethal genes to overexpress in any cell are the APOBECs. About therapeutic applications of these things. Obviously, you don't need to express these proteins for that long in order for them to have an activity, but certainly that is something that folks should be aware of when using base editor technology. That this is another, in the same way that as Trevor and I were discussing earlier, double-strand DNA breaks in lots of places in the genome, especially in the amplified region, is one sort of like sequence-based, but on another level, not sequence-based version of off-targets of CRISPR tech. I'd say the general lethality of APOBEC domains would also fall into that category.
I guess maybe I would also I totally agree with that. One thing I would add is that, I guess in addition to kind of like the systematic unintended effects of base editors, there's also, potential for random activity or things that, for example, for the APOBECs, like that match potential recognition preferences of that particular domain. And so understanding both the systematic activity of base editors, but also potential random activity is important. There's been some work from, I think, like David's lab in terms of like proxy base editing activity assays, where you basically open up R loops using something like a catalytically inactive Cas9, and then look to see whether you're getting activity from your base editor at those locations. That's one other potential important thing to look at. I think that the bar is going to be high for earlier base editing therapies because of the basis in some of these APOBEC deaminase domains that are overexpressed in cancers.
Yeah, I totally agree with Shengdar and John, and I think they did a good job covering the essays, right, for base editing of targets. Maybe I can approach from a different perspective and talk a bit more about the efforts on the engineering side that has been done to address this problem for base editors. I think that was also part of your question in the beginning. In general, I would see it as two problems, right? Like there are two legs: spurious RNA of targets and spurious DNA of targets. And I think it has been well documented by our lab and other labs that both CB and AB, I mean, as John beautifully said, the APOBEC, I mean, it stands for apolipoprotein B mRNA editing enzyme. So it's found to be like, it was known for 2 decades that it's going to deaminate mRNAs, right? So we know that they induce spurious deamination transcriptome-wide, and thousands of them. And there has been engineering attempts, mainly by our lab, secure CB and secure AB, if not abolish the complete RNA spurious activity, but really dramatically lower them. And it comes with some on-target editing trade-off, but I would say modestly. And in terms of spurious DNA of targets, which is a little more interesting because per cytosine, when you think a population of cells, there are millions of cytosines across the genome. And then when you have a population of cells, the sensitivity you need to achieve to figure out if there's a deamination spuriously happening in DNA is super hard. And I think that's why the initial efforts has come from the whole genome sequencing of I think plant, rice, and mice, if I remember correctly. And I think the reason is because when you introduce the base editor at the early stage, then you have this clonal expansion from the 1-cell stage or 2-cell stage that gives you the coverage power to actually detect them. But that these data, I would say, show that CBE can like especially canonical B format CV can efficiently induce spurious DNA editing, and then ABE is probably at lower levels, which is also in parallel with David Liu's published data and our unpublished data, where they do this cool orthogonal SA-Cas9 R-loop spurious DNA deamination assay, and then that's perfectly in parallel with the whole genome sequence. And I would also point out finally the YE1 engineering efforts of the David Liu's group. That's also very promising, along with the R33a.
I could maybe.
Sorry.
Go ahead. I guess one follow-up comment is I could maybe add a little bit more color in terms of so some of the therapeutic efforts that we're working on. We have some collaborations with Mitch Weiss's group and David Liu's group, where we're working on base editing in human hematopoietic stem cells to either induce fetal hemoglobin or for conversion of the sickle point mutation to a non-pathogenic allele. Sickle, it's called hemoglobin macassar, essentially. And so a lot of these efforts are focused on the A base editors because we think they have overall lower genome-wide off-target activity. With the sickle conversion, so the macassar project, it is kind of interesting in that we did look at a number of off-targets that were nominated by CircleSeq as a particular nuclease off-targets, and looked at them for base editing activity. And we actually did see a pretty high validation rate for the off-targets that we found. So there were some pretty high frequency point mutations that we could find as high as 80 or 90%. Kind of surprised me actually, in terms of how high that could be. We don't really have that much information on how generalizable that is, but that's something that we think that will need to be addressed.
So if I could interject, I wanted to follow up on change the topic slightly. So one of the questions for Andrew was, is Prime going to replace base editing? And I think you gave a nice comprehensive answer for that. But that's from, forgive me for saying this, but that's from the Prime perspective. And I wonder if the others of you involved in base editing might also comment on that question. Is there a future for both of these? Are they complementary? How much do they overlap? And do you expect one or the other to be a dominant method of editing in the future?
I'll give my. I think that they have pretty different use cases. I think the Prime editor doesn't obviously lend itself to large-scale pooled screens because it's even from the perspective of the nucleic acid, you need, for it to work best, you need the guide RNA itself, you need the PEG modification of that guide RNA, and then you need the nicking guide somewhere else. So those three end up in the same cell for pooled screening. Now, one could think about how to get there. I'm not saying it's impossible by any means, but I think that the simplicity of base editors is the guide, and everything else is coming from the Cas protein. I think for screening, base editing will certainly be good for going broad, but you could certainly imagine making some Prime editing libraries where the only thing that you're varying, let's say, is one codon in the I forget what you call it, Andrew, the part that gets reverse transcribed and incorporates the edit, whatever that's called. Where you have a library and you just install all the amino acids at one place. So you're sort of doing a screen of a very focused window, and that would be great for Prime editing in a way that base editing obviously just can't touch. So I think both of them will be really useful going forward, just in different applications.
Yeah, I could chime in. I agree with that, and I think Andrew had a nice summary too. I think I could kind of imagine or envision that these different technologies maybe they could begin converging more in the future. If you could target more of the pan space, then maybe the window is not as important. I think, but so maybe some of the point mutations that could be made by base editors could also be made by Prime editors, but there is definitely a whole set of mutations could potentially be made by Prime editors in terms of smaller insertions, deletions, other more complex replacements that just can't be accessed by base editing, even if you imagine all the other technological improvements. I think that to me the current state of the art is that base editing you can make certain point mutations, right? Including like, the new ones that Abraham described, and also the canonical like A to G, C to T edits. You can make sometimes you can make them with relatively high efficiency, but you are constrained in being able to make that specific subset of mutations. And with prime editing, there's probably a lot more flexibility. It seems like a really powerful technology, but the efficiency is something that's going to vary widely between cells. I guess like that's one question I've maybe posed to Andrew too, is like, are you guys thinking about what are the determinants of efficiency in different cell types, particularly important like human primary cell types that are being used for therapeutics?
Yeah, absolutely. And that's a great point. And right now, I don't think we have a full handle on that. And part of it is, I think the first thing most people jump to is DNA repair being different in those cell types. And that absolutely should be the case. But another difference is simply the delivery of the reagents to those different cell types. We have found in a few cases so far that, if you optimize certain reagents like mRNA and synthetic guide RNAs that are high quality from the right vendor, you can achieve much higher editing in these cell types that looked harder to edit when using plasmid DNA. So I for sure think one side of it is going to be just the quality of reagents and what you can deliver. The other side, I think, is DNA repair and other cell specific factors where there's going to be a lot more to investigate, and it's going to likely change from one non-dividing cell type to another non-dividing cell type. So I think it's a really rich area to explore, and I'm excited about any possible developments there that actually give us insights into engineering the system to be more efficient in those cells.
So I'm going to ask you to name
Actually,
names.
Andrew, yeah, I was just going to come to McCarthy.
It's in a challenging position.
Well, I don't want you to disparage anyone, but anybody that you've had good success with, please let us know, because everybody benefits from that.
Yeah. I guess I don't want to name a particular company because I don't feel like that's right. And things change actually, and people make improvements. So I would check with your vendor that they have good QC, that, HPLC purification is always going to help. And the longer your pegRNA, the more this becomes an issue. So maybe where single guide RNAs, you don't see as much difference from vendor to vendor. pegRNAs, we compared a few and we did see substantial differences. So if you're going to use synthetic reagents, just check with those suppliers and see if they can provide those things. And if they can, I think you'd be in a better spot with those folks.
Well said. Well said. I'm going to take a step back here and say that, and make an observation that across the sort of the history of CRISPR technology development, what we've seen is that there have been substantial increases, some data-driven, some more engineering-based. And the Cas9 library designs were evolved from a large data set, a large knowledge base on a bunch of different Cas9 guides that had been designed and used. And there was a learning from that. And so now we've got a pretty good idea of what works and why, and for a Cas9 double strand break. An engineering approach was taken by the Cas12 group in the Jung lab. So I think the general consensus was that the AsCas12a didn't work very well until the enhanced version came out, and I think that version works quite well. And so that was a targeted design effort that resulted in that. So I wonder if you guys might comment on the opportunities for further sort of enhancement and refinement of your technologies. Is it the guide designs? Is it the nuclease itself? Is it the associated domains that you've attached to it? How far can you take this, and how accurate can it get? Any of you?
I could start off by saying that I think all of the above matter. From a prime editing perspective in particular, the guide RNA design is much more complicated, I think, than most other CRISPR technologies. There's the choice of the spacer, and in addition to that, these other variables. And for all we know, there'll be additional variables added to that. So I think that's a big part of it, and maybe harnessing some larger scale screens and using some machine learning type approaches could help us design faster the good guide RNA for the particular edit. And I would say for the editor protein, I think the targeting efficiency is a huge factor. And so if we can engineer variants that more efficiently target non-canonical PAMs and increase the scope of editing in that manner, that would be really important. And for the domain aspect, finding enzymes that work in specific contexts more efficiently, and having sort of a library of these variants that you can pick from that might be suited to your particular sequence. So I think that's an area where future developments can be made.
Yeah, I completely agree. It's a huge search space, right? Recalling a paper from Lucas Dow's group where they showed just by changing the codon preferences for Cas9 itself, and then they did the same for the base editors, you could reach the efficiency. Earlier today we heard about, well, how many nuclear localization sequences do you have and where are they? What are the spacers that you use between Cas9 and what that's just a combinatorial explosion of things to try. And obviously, people tweak at it until they get something good enough to do the biology they're interested in. But there's still, for upward optimization. But, at the end of the day, those are, very trial and error experiments. And, I think that it might be hard to convince people to do that.
Well,
projects maybe.
One of the reasons I ask is that is not just for my benefit because I want the data, but also because I think all of these sort of aspects are going to be important as we transition into clinical applications and understanding both precision and the specificity and sensitivity of all of these technologies. So, if anyone has any, yeah, go ahead, please.
I was gonna say, if I can add, I've been thinking about this, genome editors for some time, and I think obviously we're all very excited about CRISPR-Cas genome editors. They're like really powerful RNA-guided technology. I started working on this intersection between zinc fingers and talons, and one aspect that was pretty interesting about talons is that you can install double-stranded break at any position really in the human genome because of the density with which they can be designed. So thinking about the empty spaces, I think being able to access any base, with CRISPR-Cas nucleases by expanding the targeting range is still, it's a problem that's been worked on for a bit. There's been many improvements, but there's still a ways to go to be able to access any base. Being able to bind to any particular base could also open up opportunities for base editing, right? Being able to particularly make point mutations in any particular position. And then on top of that, I think being able to make more complex types of edits, whether they're smaller insertions, deletions, or the insertion of large cargo. I think these are all areas that are very open and in terms of what needs to be worked on in genome editing technologies.
So I think I would like to touch base, continue maybe off target and then library screen topic. So we know a CRISPR could introduce small deletion or large deletion. Just wondering will that affect the off target analysis, or how does people use those library to look in this perspective if you have small deletion or large deletion in your library screen? And also from base editing or prime editing point of view, has people seen use the base editor or the prime editing, you seen those small, large to decent size of deletion?
Maybe I can quickly mention something about that. That bridges between the technologies and the larger deletions that people usually don't check much. We have the Unitas technology, right? That looks for larger deletions and stuff. But I feel so while I'm a big fan of the nuclease Cas9, for example, I envision field converging more and more towards base editor and prime editor or any technology that doesn't really introduce double-stranded break. Just because I feel based on this preprint by my archive paper, David Pellman of Harvard Medical School, where they actually kind of showed that chromotripsis, that's what fancy name of it, chromotripsis, can be an on-target consequence of a Cas9 nuclease cleavage. So essentially, when you cleave a chromosome arm, the telomeric part of it can actually, at quite high efficiency, it's 20–30% of time, can just float around freely and diffuse. So the problem is, I guess, we've been focusing a bit too much for the small indels and due to technological limitations, but sometimes we easily oversee the or undermine the value of simple karyotyping and fish and so on and so forth. So to this end, yeah, I just urge everyone to read the paper. But while I'm a big I'm super happy about the findings of course, the Vertex and CRISPR Therapeutics. And we know zinc finger nuclease has been in people following transplant of hemoglobinopathies. But at the same time, it's a bit, let's say, interesting. I mean, we need to probably check more about the big structural variants as well, like you mentioned, the large deletions.
Right. And I think that's where it's really important to make sure that we're not talking about, what one intends to do is really important, right? If you have a 0.1% chromothripsis rate in hematopoietic stem cells that you're going to put into a patient, that's one thing. If you have some genetic screen in K562s, who cares? It's not going to drive the signal of your screen in any meaningful way. So I think, we want to be aware of these observations, no doubt. But at the same time, I don't think we need to revisit all prior CRISPR results with the idea that they were, some false positive. You were saying by any means, but I just want folks to be clear that we want to be tempered in our language.
Yeah, I apologize. Let me clarify. I didn't mean that they are false positive. I was just saying the off-target analysis that are done, of course, are carefully vetted. But I would say maybe the last few years we've been focused a bit too much on the smaller single nucleotide variations or
the detection challenge is much bigger. Yeah, absolutely.
And I would echo. I agree with that point. That so it's not just about the small indels, right? Like I think it's about what are the overall changes that you're making to the cells? Like could there be large deletions, inversions, translocations, and things like that? I think with the Hellman paper, I think the important thing to note is that they did detect chymotrypsis pretty shortly after editing. But one question is do those cells survive over more than a few days, right? Those cells I think typically seem to go away. I think in their paper they couldn't detect it after a week. So that's another thing to keep in mind therapeutically.
I mean, the one thing I'd like to include here is, we're talking about cultures and that are carried over for several generations, and there's always the possibility that some degree of evolution or something has arisen in your cultures that you don't know of. So kind of a universal look at the genome stability before you get too far down the road, I think is usually a good investment.
So thanks, Steve. I wonder if you have any comments on any technologies in the pipeline or any preferred approaches that you guys have for how we can approach this problem?
Well, I kept waiting for somebody to leave out any of my favorites, but they've all been brought up in conversation as you might expect with a panel like this. I mean, the vast majority of technological approaches that we see to on and off target validation are targeted sequencing, and much of that comes down to how you pick the off targets that you're going to look at, and so those computational approaches to look at where the guide RNAs are likely to interact, I think are critical. So the guide-seq, circle-seq, and digenome-seq are probably the ones that we see most people utilizing to the greatest extent. I would also put in I like to see some RNA-seq and to see are the expected things going up and down, and are there spurious things coming up and down, which I think is usually interesting just from a biological and understanding the mode of action of the edits you're doing anyways. But I think that's a really important part of making sure that you're not getting 10,000 genes that you didn't want turned on